first sparks on quiet streets, two hearts finding where to meet. But life pulled us apart, left me talking to the dark. If I called you tonight, would it feel the same? Or have we both moved on to different names? Take me back to the start, before the quiet and the scars, when the world felt small and your hands felt warm. Take me back to the start, if only for a moment. I've been up since 3, scrolling back what we used to be. Half truths in this late night chat, both of us know.
Welcome to Let's Get to the Marks. Okay, preparing for the bio exam tomorrow. So, yeah, we're going to go over bio and get ready for bio. So, yeah. Here we go. What? Yeah, right. Where are we? What we doing? Yeah, we're gonna do just a couple of random multiple choice questions just to start. So these are on just some random topics that are coming up just to get us warmed up. Then I'm gonna go through the syllabus. I'm gonna stick to the AQA Biology syllabus, but if you're doing Edexcel or OCR, there are some things that will be relevant to you. There are, for instance, the first part of the live will be going through cells, microscopy, mitosis, cell division. That's going to be relevant to you. And also when I cover photosynthesis, respiration, maybe not photosynthesis, but respiration is definitely part of the OCR, what you're doing as well. However, there are certain topics that you do in paper 1 that are in AQA paper 2. So I'm sticking to the AQA Biology and Combined Science syllabus, but there are some bits that will be relevant to you. You can have a go, guys, at these 5 questions. So number 1 is an embryonic stem cell can differentiate into any type of cell, differentiate into only one type of cell, can only be obtained from embryos, only produce haploid cells. And then we got question 2, the term used to describe the number of chromosomes in an egg or sperm cell is diploid, gamete, haploid, zygote. Then we got number 3, which term describes the area of a root where many cells are dividing by mitosis? Is it meristem, root hair cells, xylem, phloem? Or number 4, which I can't see without my glasses, get the old specs on. The cell is dividing by mitosis. Which structure is labeled Z? Is it an allele, a chromosome, spindle, or nuclear membrane? And then 5, which process occurs causing the divided cells to become specialized? Is it meiosis, cloning, differentiation, cytokinesis? Put your answers down in the chat if you want to have a go. Okay. Yeah, so some of you put A, well done. What's 2? It's C, half the amount of chromosomes. What's 3? Hopefully you got 3 is, no, 3. Look, that is to trick you. The root tip, which contains meristematic tissue. So is the meristem is the correct answer for 3A. The root tip contains meristem. Root hair cells are specialized cells that are for absorbing minerals and water by osmosis and minerals by active transport. Right, water by osmosis, minerals by active transport. So 3 is A, 4 is B. Anyone go for 4B? 4 is B, that is right. Quite a couple of people put 3 root hair cell, 3B. It's not 3B. There is meristematic tissue in the root. When we do the mitosis practical, we squish a root because we want those meristem, we want the stem cells from the tip of the root, but the root hair cell is something different than the tip of the root. Okay, which process occurs, causes blah 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 blah. The 5 is C. Differentiation is how cells become specialized from a stem cell. Anywho, let me just hit you with another 5, and then we'll actually get on working through the course. Let me hit you with another 5 just to warm up your brain, keep you warmed up. Which word describes an organism that causes disease? So you got to know this. If you're doing AQA, you got to know that. For paper 1, you don't have to know that for all of them, but yeah, it depends which exam board you're doing. 7, which data can be used to calculate BMI, body mass index? And then we got which part of the blood transports insulin to target organs? Is it the plasma, the red blood cells, white blood cells, or platelets? Number 9, the organelle labelled X. Huh? I can't see no organelle labelled X. Controls the activities of the white blood cell. What is the name of the organelle? We better skip number 9 because I must have cut off the image. What an idiot! Let me show you. I'll draw it. Okay. So this is what the X was. So I'm doing it, if I remember right. I must have cut it off. What a wally. So it's a white blood cell, and part of it is labelled X. What is the part labelled X? I know. Easy peasy, right? Which part of the blood causes the blood to start clotting? Is it erythrocytes, lymphocytes, platelets, antibodies? Let me check your answers. Oh, Lucas has put up 6A, 7C, 8C, 8A, sorry, 10C. So 6A, yes, it's a pathogen, Lucas. Well done. You said 7C, mass and height. Absolutement. What is it? Is it mass over height squared or height over mass squared? One of the two. I think they give you the equation in there. Which part of the blood transports insulin? Insulin's a hormone, guys. So I can see some people, Lucy's put C. Yeah, I think that was for its plasma. Yeah. Sorry. 8 is A. Holy shmoly. The plasma transfers urea, proteins, hormone, which are like hormones, carbon dioxide, vitamins, minerals, water, salts. You can like name 50 things. Okay, number 9. This is supposed to be a white blood cell. I've just mashed up my screen a little bit there. Why did I do that? This is a white blood cell. Now, watch out for this. Don't let this put you off. You don't have to know this on your exam board, but this is called a multi-lobed nucleus. You don't have to know this on your exam board, but that doesn't mean they won't put it on an image just to like mash with you. It's the nucleus, okay? That's the nucleus of a white blood cell. The phagocytes that actually engulf and digest have a bit of a weird looking nucleus. Some of them do. Some of them have this multi-lobed nucleus. The ones that make antibodies don't. They have a nice spherical, perfectly round nucleus. Which part of the blood causes blood to start clotting? The answer is 10D. No, it's 10C. It's platelets. My brain. Oh my God, that's not a good start, is it? Platelets. Nice. Awesome. Okay, let's move on. Let's get started. What do we want to start with? We want to start with cells. And I'm going to just work through the whole course. So it's going to take me about 3 hours. I know, long, right? Okay, nice and easy peasy. Lemon squeezy. Let me just test you lot on this. What structure is A and what is its function? See if you can put up the structure. What's the name of this structure and function? And then let's go with B next. Structure and function, folks. I'm going to call that B. B. And then this is C. Make them stand out on the camera a little bit more. Let's make that D, although my arrow's a little bit bent. And then let's make this E. Structure and function of each. You should know them all. Dead easy peasy, just warming your brain up. Mitochondria. Lucy, you got to be specific. There's only one specific chemical. There's more than one, but there's a specific chemical reaction they want. It's mitochondria. And they want a very specific reaction. Close, Zain Mahmood. What type of respiration? Oh, someone above you put that. Nandana, it's aerobic. Forgive me, I'm looking at you guys on the chat on my little phone because I got this on my screen, so I may get your names wrong. No offense meant. Aerobic respiration for A. This is the cell membrane. Controls what goes in and out. Am I going to be able to write this all in? It's going to look pretty rotten, isn't it? Controls what goes in and out. Now that's very basic, so controls entry to the cell, controls what can go in and out. Then we got cytoplasm. Now depending on your exam board, they might change the meaning of this, but it's where chemical reactions happen. I know what you're thinking. Hold on a minute. Isn't aerobic respiration a chemical reaction and that happens in the mitochondria? It's not where every chemical reaction happens. It's where most of the chemical reactions happen. So anaerobic respiration happens here. That's why specifically we're specific about aerobic there. Anaerobic does not happen in mitochondria. So cytoplasm, it's where chemical reactions happen. Now, depending on your exam board, you may have to include it contains enzymes. Oh, I can't fit in where chemical reactions happen. It's all gone a bit pear-shaped for me here, guys. That doesn't make sense. Okay, ribosome. Hopefully you know that is protein synthesis. I can't spell ribosome. This is going to get messy. Don't write all of this down. It's too late for that. But if there's one you don't know, just make a bada bada bing. This controls the activities. Do not say the nucleus is the brain of the cell. No one's giving you marks for that, right? A brain is a complex organ made of tissues and millions and trillions of cells. It's nothing like the nucleus. The nucleus controls the activities of the cell, but more easier to get the mark is to say it contains DNA, and that just wraps that all up. Oh! Right, controls activities of the cell, we can say that. Where DNA is stored, either one will get you that. Now, of course, I've not left any room because I'm a Wally. Let's go A, B, C, D, E, and then we got F. Obviously, A, B, C, D, and E are all in this as well. But what's F, G, H? Now, there's going to be some students, F, G, H, A, B, C, D, E, F, G, H. Yes, that's right. Right, there are going to be some students that mislabeled this one as nucleus. They're going to go for the biggest round thing in the cell. Because when you're dealing with an animal cell, the biggest thing's the nucleus. But in a plant cell, there's something bigger than the nucleus. Remember that. So chloroplasts contain light, absorb light for photosynthesis. Nice. Chloroplasts, chlorophyll, you get the coup. I don't think I can fit it in, guys. So I'm just going to... chlorophyll, G, cell wall. So have a look at the chat. You'll see people putting in the chat the correct thing. Oh, that's not chlorophyll. Gosh, see, it's all gone Pete Tong. Chloroplast. Right. Chloroplast contains chlorophyll. Chlorophyll is the green pigment, the chemical that absorbs sunlight. It's found in the chloroplast. G is cell wall. 10 points if someone can tell me what is in the cell wall. Thank you, 52VV, for telling me about the vacuole and that it contains cell sap. Yeah, it also regulates, it helps support the cell, due to osmosis and stuff. It helps regulate stuff. But it contains cell sap. You'll get the mark for saying that. Some teachers are fussy and they want you to put permanent vacuole. Normally they'll allow that at GCSE. What's the chemical? Cellulose. Boom! Well done, guys. Yeah, cellulose is found in the cell wall. Oh my days. That is so disgustingly messy. Yeah. Okay. Any questions? Does anybody have a question about any of the cells? Obviously not about my awful writing or anything. In fact, sod that. Let's put it up so it's nice and neat. Ta-da! There we go. Right. I don't like my labeling of the mitochondria on this one, but there we go. If you need to make a note, make a note. What do we call animal, plant, fungi, protists? 4 different kingdoms of life: animal cells, plant cells, both multicellular organisms. But if we look at their cells, if we look at fungi cells, which can be single-celled or multi-celled, and then protists, which are only single-celled, if we look at those 4 types of cell, they all have a nucleus. What do we call those cells? Eukaryotes or eukaryotic cells. Lovely jubbly, well done. What do we call these types of cells? Eukaryotic. Oh my gosh. Eukaryotic cells. Lovely. Excellente. So, well done, guys. Any questions about cells? You don't need to say permanent vacuole. Yeah, I'm going to be about 3 hours, but you know, I would, yeah, science isn't till the afternoon. You can watch some of this on your phone or something on YouTube if you know, have your phone charged. It'll be up to watch for a few days. Not that you'll need it after tomorrow. You can burn your biology books unless you're doing A-level biology. Actually, don't burn your book. You've got B2 to do after half term. Okay, let's zing on. So, was there any questions? I think it's better because, yeah, some animal cells do have a vacuole. Yeah, white blood cells can temporarily have a vacuole when they engulf and digest pathogens. But you will not get extra marks at GCSE generally. So, you can just call it a vacuole. It's at A level that you have to say permanent vacuole. What do we call cells that do not have a nucleus, guys? Prokaryotes. Look at you guys flying. Awesome. You're ready to smash it up tomorrow. Prokaryotic cells or prokaryotic cells. And what do we call tiny circles of DNA? Anyone know? They only contain a few genes. Got a special name for them. Plasmids! Awesome! Right, these are tiny circles of DNA, and when the bacteria reproduces, or the prokaryote reproduces, by the way, do you remember how prokaryotes reproduce? Anyone remember how prokaryotes reproduce? Oh, don't tell me I've got to kick people out already. You know what? I think last year I had some real lunatics in the house. Binary fission, not mitosis. Although, check your exam board. If you're doing AQA biology, you need to call it binary fission. Now, binary fission, look, I've got let's put 4 plasmids. Binary fission, the wiggly shoelace looking DNA. Bacteria have like a shoelace DNA. By the way, what do we call that DNA that looks like a shoelace? This one in here. Do you know what we call that DNA? Loop of DNA. You can call it loop of DNA or chromosomal DNA. Cool. Or chromosomal DNA. Remember, you can, if someone's getting on your nerves in the chat and you're like, oh my gosh, this person's winding me up, remember you can left or right click on their name and you can blank them out the chat so that they appear for other people, but anything they say you will not have to worry about. Circular loop, chromosomal DNA, loop of DNA. Great. Now that's going to be identical, but check what happens with the plasmids. I mean, I don't think they'll ask this, but it is on the spec. The plasmids do not have to separate evenly. So they're not actually, although we would normally say they're clones and they're identical, they're not really, because these might have 2 or 3 more genes than this one. So that's me just putting that there. I don't think they've ever tested it, but you never know when it could be the day. Now, the cell wall of bacteria, what's the difference between the cell wall, oh my gosh, of bacteria that was foul. What's the difference between the cell wall in bacteria and that of a plant? Does anyone know? No cellulose. Thank you, Lucas. Yes, I'm sure other people put that as well. But yeah, no cellulose. So if you're asked for differences between a bacteria and a plant cell, yes, a bacteria doesn't have a nucleus, a plant cell does. They both have a cell wall, but the bacteria cell wall does not contain cellulose. It's going to be triple and combined. I'm going to cover it all. I'm going to cover therapeutic cloning, absolutement. Yes, yes, yes. Is anyone being a pain in the butt? I only subscribed to violate you. Oh my gosh. Yeah, you always get some losers. You know, all I can say is you always get some proper hardcore losers in the chat. You can't do a lot about that, guys. you can't do a lot about that. What we'll do is we'll give the dude a ban if he's acting like a dipstick. We'll give him a little ban. Yeah, I haven't made anyone thingy me bob. Oh my days. What the ass? Nice, nice, nice. Oh, you see, the problem is then I've got to go through and find the person. Chase! Oh, there we go. Let's stick them on the old. Let's remove. Now, what we want to do is we want to put user in timeout. Let's give them a 30-minute timeout. There we go. Got rid of that person for 30 minutes because they're some sort of loser. So, like, sometimes you're going to get that. You just have to, yeah, they're just going to have to hold a little ban.
And it's going to be a card for the Liverpool captain, and it's red.
Deco, that's his second yellow. He's off.
Yeah, he's gone for 30 minutes. So, yeah. Keep it respectful. Right, let's go, let's go, let's go. Yeah, I will cover therapeutic cloning. Okay, let's go, let's go. So, which cell structure contains DNA? Problem is, with some of these people, I will make a couple of people admin, but, you know, don't feel like you've got to hang around if I make you admin at some point. There'll be people that have been in the lives previously because, like, over several live, I have had it where, you know, you get someone who goes in the admin and then they get like power and they just start acing everybody out of the thing. So yeah. All right. So here's some 10 questions. You can just test yourself on that, and then we're going to move on to microscopes. I might just make while you guys have a little answer on that. Let me ba, 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 ba. There you go. Ishal's admin. If someone's annoying her, she can put them on timeout. She's a member as well, so you know anybody who's a member is going to be instant admin. So anyone's rude to them, they can just dash them. I'll make some other people admin later on as well. Okay, let me get on. Let me get on. Okay, here are the answers for those. I'm out of nucleus. Let me just check I've written all the right answers because it wouldn't be my first time. Organelles, respiration, name of cell structure found in plants and bacteria but not animals. Cell wall. Name 3 cell structures found in plant but not in animals. Cell wall, chloroplast, vacuole. What is a plasmid? In which cell do we find them? Small circular DNA, bacteria cells. Which of the 3 cells looked at is the smallest? Bacteria. Bacteria about 1,000 times smaller, guys. So that is a difference that you're allowed to say in exams if asked for a difference. What name is given to the cells that have no nucleus? Prokaryotes. And which chemical is in the chloroplast? Oh, right. Don't worry, I will add some other people to the old. I will add a few mods as we go through. Okay, what do we call the ability of microscopes to make an image larger? Hopefully you guys are all over that. You know that. That's called magnification. So all microscopes can magnify. Light microscopes go to, oh my days, light microscopes go to about 1,500. Electron microscopes can go up to like 200,000. So they have a much bigger magnification. But name an organelle you can view with a light microscope. So you can see chloroplasts. They're pretty big. You can see the nucleus. What you can't see is ribosomes. So you can't see ribosomes or plasmids. So I'm going to put ribosomes and plasmids here. By the way, organelle, you don't have to know that. That's not on your syllabus. I know they use it a lot. You can say cell structure, okay, or subcellular structure. Ribosomes and what else did I waffle about? Plasmids. You can't see these with a normal microscope in school. What are 2 advantages of the electron microscope? So we've already said one. Magnification. And you've got resolution. What does resolution mean? Higher resolving power, thank you, and magnification. It's how clear the image is. Good. How much detail you can see. how clear image is, you can see smaller objects as well. So you could see ribosomes and things like that. How much detail you can see. There is a specific method for that. Right, let me just go through very quickly as I'm going through everything. So some of you might be like, oh my gosh, I can't be bothered to watch all of this. Then you can jump out and jump on. But I'm going to go through what you got to do. So there's a core practical to do with any questions about microscopes before we move on. There is a yes, that's right. The ability to differentiate between two objects and see them as separate. Probably won't help with your uni degree, I wouldn't have thought, Spaniel Daniel. Unless your uni degree is art, and then you can look at my artistic skills and take some tips. Right. First part of a core practical, you're going to be asked how to prepare a slide. So potentially you could get asked how to prepare a slide. Comes in 4 steps. So if you don't know it, get to know, because it could come up tomorrow and then you'll be like, whoa. So you're going to put a water drop on the slide. Then you are going to, so that's the first step, is place drop of, obviously if you don't know what that rectangle of glass is called, it's called a slide. Place drop of water on slide. Then what we're going to do is float. We want to float specimen. This is getting a bit long, isn't it, doing this? Right, float thin. Why does it have to be thin? I'm calling it specimen on water. What are we doing next? Yeah, you don't have, I mean, are they going to take a mark off if you don't say put a drop of water on the slide? Not necessarily, no. It just adds moisture and whatnot. And then we're going to add stain. So then we add stain. Now, there are different stains. I believe on the AQA syllabus you will get the mark for just saying stain, right? Add stain so we can see details more easily, whatever. You could just say iodine, there's methylene blue, there's toluidine, there's eosine, there's just like millions of stains. You don't need all of that. And then finally, what do we call that tiny bit of glass that you're going to push down, gently push down? Does anyone know what we call? Place the cover slip at an angle, push down to avoid air bubbles forming. Absolutement, 100%. So push down, cover slip. I'm going to say gently. And I've run out of room. But we push it down gently to remove air bubbles. If we leave air bubbles, they're known as artifacts. Artifacts, not like interesting artifacts, like, ooh, they're like, they get in the way and they obstruct your view. The amount of times people say, oh, sir, look at all these cells. I'm like, no, they're air bubbles. So you push down the cover slip. The cover slip is a very fragile piece of glass. You push down gently. If you push down hard, you will split the cells open and leak their cytoplasm out. Not that you can see it, they're microscopic, but you're going to smear them and you're not going to get to see anything. So I don't think I've missed out anything. This would be like, you probably, you could say place the drop of water on the slide. Use a scalpel, sharp knife to cut a thin slice of your onion or whatever. Hey, safety. Yeah, safety. They might ask you, like, use a chopping board when slicing stuff. Use a scalpel to make sure the onion is thin. Why do we want thin onion or one layer of cheek cells if we're looking at cheek cells? That's so the light can go through. Add the stain so we can see details. Push the cover slip down as well. There we go. Bish, bash, bosh. Right, let's move on. The next bit that we have to do is how to use our light microscope. So here's our light microscope. Just see if you can name these, then you're probably halfway there. This is A, we got B, we got C, we got D. This is also a little turning thing. I'm going to call that E, and then F. There is an arm there as well that connects it all. Yeah, you can put in the chat if you can name them, and then I'll write down how to use it roughly. We don't want to spend too long on it. There have been some really disgusting hard questions on microscopes before, but pretty much normally you can. Okay. I'm not 100% sure microscopes will come up, not 100% at all, but you know. I don't have any predictions.
What's your strategy?
Don't need any.
What's your prediction?
Prediction?
Yes, prediction.
Pain.
Yeah, I'm not 100% sure if microscopy will come up, but it is, you know, a common topic. So I don't deal with the predictions, as you can see. I just have stupidness. Right, so here's how I'm going to say to use the microscope. So the kind of question, how do you know whether to talk about the slide, the drop of water, the iodine? They'll say, how do you prepare a slide? If they want you to just talk about the microscope bit, they will say, you have a prepared slide. How would you use the microscope to view your prepared slide? So this is the bit I'm going to go through. Oh my gosh. We're back. Right. Pen. Behave. Okay. So what I'm going to do is I'm going to say clip slide to stage. So again, do you have to have every single point? No. Like there'll be 6 or 7 points on the mark scheme if it was 4 marker. If it's a 6 marker, there'll be like 8 or 9 points. You just need to get 6 separate points. So clip the slide to the stage is a dead easy one, right? By the way, C is the stage. I know you know that. Then what I want to say is we're going to raise the stage. But before that, I'm going to say, look at me, select the lowest. It doesn't matter too much which one. Select the lowest objective lens. Oh my days. So objective lens is this one. You're going to select the, I can't speak. Select the lowest power objective lens. I'm trying to shorthand everything. Clip the slide to the stage. Select the lowest power objective lens. Raise the stage until lens almost touching. How do you raise the stage? You're going to turn knob D, and this is the rough focus. No. Over here, I've got the fine focus. Over here is the stage, and up here we got the eyepiece, which I've just, written where my face is. So I'm going to say clip slide to stage, select the lowest power objective lens, raise the stage until almost touching. Now I'm going to say look through eyepiece. Oh my gosh. Look through the eyepiece. And then I'm going to say slowly lower the stage with the rough focus. Turn rough focus to lower stage. So you're starting like this with your eyepiece, like, oh my gosh, you're starting with your eye looking in there, and you've got your stages right there. Okay, as time goes on, this is going to come down so that you're looking like that and you're lowering it. The stage is coming down away. The reason we do that is for safety. If you start with the stage down here and you rise it up, you can smash the glass. If you're while you're looking in the eyepiece, you can't keep your eye on the stage. It's impossible for you to smash the glass because you're lowering it away from the objective lens. So the metal bit of B is not going to crack into the slide on part C. Okay, so we turn the rough focus to lower the stage. Once we can see image, now we swap knobs. Okay, that was a bit no diddy. Now we swap knobs and we go to the fine focus. So we now use fine focus to make image clear. So the fine focus is for making the image clear and sharp, detailed, however you want to put it. And now, finally, we select what power we want on the objective lens. Select desired power on objective lens. Oh my gosh. I didn't mention the light source, which is F. I couldn't be bothered. So rare that you see the light source on the mark schemes. So there you go. You can see I've made 1, 2, 3, 4, 5, 6, 7, 8 points. You only need 6. And there's probably other stuff I haven't said. Now, just be aware if they say they want you to get a times 50 magnification. Sometimes the eyepiece lens has a magnification, like that might be times 5. If you select times 100 on there, your total magnification is going to be 5 times 100 equals 500. So just be aware that you can multiply the eyepiece lens by the objective lens, you'll get the total magnification. Coolio! You don't have to say sharpen the image. You can call it coarse knob. You can call it the rough focus. You can call it the coarse knob. You can call it the adjustment knob. You can call it different things, okay? So you can call it the rough focus or the coarse knob or the adjustment knob. You can call it different things. Both will work. Okay. The lowest power on your objective lens depends on what microscope you're on. So some microscopes it's times 4 is the lowest power, like will be the lowest kind of power. Some microscopes, light microscopes, can go all the way up to times 1500. Yeah, that's right. It's like times 4. Turning it on, yeah, you won't get a mark for turning it on or taking the cap off the eyepiece or something like that. Anywho, last thing I want to go over with the microscopes, and then we can move off on microscopes because it's painful. The last thing I want to look at with microscopes, okay, is... Yeah, I ain't doing that. Right, use a sharp pencil, make unbroken lines, no colouring or shading, label structures with straight uncrossed lines, and write the magnification. So I'm just going to highlight these errors because you might not know about these errors, so let me highlight these for you. Nucleus, this person should not have coloured in the nucleus. That is a mistake. Good that they did this. They put times 500 magnification. Excellente. Now let's look at the sperm cell. They crossed their labels. You must never cross your labels. That is bad, bad, bad. But good that they didn't shade in the nucleus. Well done. And they wrote the magnification. Now let's look at this bottom one. No mag. So they need to write magnification. By the way, also, you can't do this. This would not be correct. That is a broken line. You're not allowed broken lines. That would be... You're not allowed crossed lines on your drawing, so you can't do that. Like, oh, that's my cell membrane, right? That is bad because, look, the line is crossed. So don't cross over each other. You get the idea. Sharpen your pencil. No colouring or shading. Orthogonous. That is AQA. You need to know about this. Millimeters to micro. Yeah, it's 1,000 for each of them. So if you want to know about the scale, let me zing down here and I'll give you a little rundown on the scale. Let me give you a rundown. I see some people in the chat chatting about the old microscope scale. We will do a microscopes question in a sec, I promise. You can have a go at this question. If you're not interested in what I'm waffling about here, you can have a go at this question and test your skills. So, oh my good gosh, where are we? Where are we? We are here. Bosh. Okay, so Let's start with meters in the old metric scale. The great thing about the metrics, back in the day, they used to use something called the imperial system of measurements, which was great for craftsmen and tradesmen, people who chop things up, you know, like feet, inches, yards. That was fantastic, because if you're a furniture maker, it's much easier to chop up a 12-foot bit of wood into 4 even bits, right? You just chop it into 4 threes, but you can't chop a 10-foot bit of wood into 4. You got 2.5. It starts getting hard for craftsmen. But the metric system is much better for scientists. So we use the metric system, and the imperial system is out now. So 1 meter. If you go down to a smaller unit, this is the bit that mashes up people's brains. If I want to turn 1 meter into millimeter, I times by 1,000. By the way, I don't need to put the 1 there. I don't know why I'm putting the 1 there. Let's get rid of the 1. We can just assume that's 1. Bosh, bish. So this mashes up people's heads, because they're like, but sir, why are you making it bigger by timesing by 1,000 if it's getting smaller? Millimeters are smaller. Yes. So think about, if you're getting confused, think about your height. You're like 1 meter 6, 1.6 meters. How tall would you be in millimeters? You'd be like 1,600. So when we go to a smaller unit, you go bigger. Now, if we go from millimeters, the next stop is micrometers. I can't do the funny Y thing on that, so I'm leaving it like that. We're going to times 1,000 again. So there's actually 1,000 times 1,000. There's 1,000 micrometers in a meter. And then the next stop is nanometers. And then guess what? I've run out of room. The stop after that is picometers. Now, if you're going up in the other direction, if you're given nanometers and you want to go to micro, you divide by 1,000. So each time you jump up, you're going to divide by 1,000. If you're going from millimeters to meters, you divide by 1,000. You get the idea. It's counterintuitive. On the way down, you multiply. On the way up, you divide. So it's kind of, they call that counterintuitive. It kind of goes against the way the brain normally thinks. Oh, we've got some messages being deleted. Hui. The respiratory system. Yeah, I will be going through the respiratory system when we get to organisation. I'm going to be on for 3 hours, so I'm going to get through the whole of the AQA Biology course. Hey, Ishal, don't you stress. You've got your exam tomorrow. Don't stress about kicking anyone out. You can leave the. I will get round to that at some point. I'll put some questions up and I'll kick people out. Don't let that stress you out. Oh yeah, centimeters to micro. That's a good point. Centimeters. I always say get to millimeters first. So convert your good note. That's a good point. Someone put in the chat there. So convert centimeters to milli first, and then you can do the 1,000 scale. So just by times 10, right? So if you're given 2.5 centimeters, just put it straight into millimeters, 25 millimeters times 10, and now you can use the 1,000 scale, like by timesing by 1,000 on the way down and dividing on the way up. 2 similarities between prokaryotic and eukaryotic cells in the diagram. First of all, which one of these is the prokaryote? My stupid board. Oh, let's move that over there. My stupid writing board. I have no idea what would be better for you. Prokaryotic cell. These two are eukaryotes. Eukaryotic. This is a plant cell, mesophyll cell, so like spongy or palisade, and this is a liver. So I'm going to go through the whole course today. Give 2 similarities between prokaryotic and eukaryotic cells. So 2 similarities. What are 2 things that all cells have? And you should be able to go, all cells have cell membrane and all cells have cytoplasm. So yeah, great. That's 2 similarities between all cells. Give 3 differences between the prokaryote and these eukaryotes. Well, you can see the size difference, right? 30 times bigger. And what is that? Well, if I divide by 1,000, I'll get the micrometers and that whatnot. But yeah, that's millimeters. Sorry, if I times by 1,000, yeah. So that's way bigger. That's times that by 1,000,000. Yeah, that's way bigger than that. So what we can say is prokaryotic cell is smaller. And you can word these. Make sure, one mistake some students do is they don't tell the examiner which one they're talking about. So make sure you tell the examiner which one you're talking about. So always start your sentence with prokaryotic or eukaryotic. So prokaryotic here, I can say prokaryotic, no nucleus. Nice. What else? Now, I'd like to say the prokaryotic has plasmids, but look at these little gits. They've drawn plasmids in this mesophyll cell, what look like plasmids. So I'm going to steer away from that. What else can we say? I can say it has a cell wall with no cellulose, maybe. Cell wall with cellulose. They might let me have that, with no cellulose in it, because that's a difference. Has a cell wall, but it doesn't contain cellulose. Oh, what about chromosomal DNA? Yeah, I should stop with that. That's a better one. Prokaryotic has cell wall with no cellulose. But I could also say, I'm just going to put quotations there, has loop of DNA or chromosomal DNA, loop of DNA. And then potentially I can say it has smaller ribosomes, but you don't need to know that at GCSE, but the ribosomes in prokaryotic are smaller. Okay, it could have a flagella. That's right, cat aviation. Prokaryotic don't have mitochondria. Thank you. They do have ribosomes. They do have ribosomes. Look, those little black dots are ribosomes. Everything has ribosomes. That's another one that they both have. They both have ribosomes. But no, I'm going to say prokaryotic has no mitochondria. Absolutely. In fact, mitochondria are about the same size as a bacteria cell, so they wouldn't fit in. One of these is the same size as that, so you can't fit a mitochondria in a bacterial cell. Okay, what are we on here? What are we doing? What are we doing? What are we doing? Okay, we're going to get off cells in a second, guys. I'm going to do a couple more questions on cells, then we're getting off of cells, and we're going to move on because this is taking too long. Biology AQA. Explain why an axolotl may die in water with a low concentration of oxygen. 4 marks. Nice, yeah. Okay, yeah, so I'll match these up for you. So, stage. Hey, that's where we put the slide, isn't it? Objective lens makes the image bigger. It's the focusing knob, or the coarse adjustment, or rough focus, moves the lenses up and down to the specimen. It can move the stage. Something moves the lenses or the stage. Depends on your microscope. Lamp shines light so it can be seen. Okay, whatever. Total magnification of the microscope is 400. The magnification of the eyepiece is 10. Calculate the magnification of the objective lens. So total mag equals eyepiece mag times objective mag. So what we're saying is 400 equals 10 times something. So it's going to be times 40. Hope that was all easy peasy pudding and pie. Right, let's move on. Okay. You know what? Let's move on, otherwise I'm going to be forever. I'm taking longer. This is the problem. I always take longer than I should do. All right, who can tell me 2 places where stem cells are found in animals? What are the 2 places stem cells are found in animals? Oh my days, I wanted to feel that. All right, guys, where are stem cells found in animals? Bone marrow, boom, and embryos. Good. Bone marrow and embryos. For GCSE, that's all we have to know. Don't go telling me, oh, sir, they're found here and there. They are found in lots of other places. But embryo and bone marrow, what's the difference? Which one can become any type of cell? These, embryo, any type of cell. I mean, that is some crusty writing. Bone marrow, only a few cells. Okay, so this would be embryo ones because they can become nerve cells, muscle cells, blood cells, liver cells, intestine cells, any type of cell. And we call these specialized cells, these ones here. Excellente. Do we really want to go through all of this? It's long. Right, I want to get off this topic. How long am I? I'm going to be on till 10, guys. That's the reality because I'm taking an hour over the first unit. The first 2 units are the longest units, to be honest, though. They are the first 2 units are the longest. Good. Right, sperm cell. How's it adapted for swimming? Just test this in your head. I'm going to put the answers up rather than waffle through it, you guys. How's the sperm cell adapted for swimming? What is the job of a sperm cell? Why are there lots of mitochondria in the tail? Why are there enzymes in the head of the sperm cell? And then for nerve cells, what is the job of the nerve cell? And then 3 marks, how's a nerve cell adapted for its job? You need to know that. It could come up. Muscle cells, what is the function of a muscle cell? What does the muscles do? And why are they long? Why do they have lots of mitochondria? Yeah, I've got to do that. How did Blue Rook Chess ever get back? He's like a slippery snake. Okay, how is it adapted for swimming? So I'm going to put the answers up for these because you guys, I'll take too long otherwise. So here are the answers. The sperm is streamlined, its head, so that it can, less friction, it can move through liquids easily. It has a long tail, which it can move. It has lots of mitochondria to release energy by respiration so that it can move its tail for swimming. The job of the sperm cell is to bring the male DNA to the female DNA. Don't forget the sperm cell has haploid number of chromosomes. And the enzymes, I haven't put the answer up there for enzymes, but the enzymes are in the head to digest the wall of the egg. So it has protease, lipase, amylase, these enzymes in the head of the egg, the head of the sperm, and they're released. It's a part of the sperm called the acrosome. You don't need to know that. What is the job of the nerve cells? They carry electrical signals. Okay, so they carry electrical signals. Don't say electricity because they might not accept that. So say electrical signals. Okay, how are they adapted to do this job? They're long, they're branched, and they're interconnected, or they form a network so that the messages can go all over the place. Muscle cells can contract. They're long so that you can get big contractions and a lot of force, and they have lots of mitochondria to release energy by respiration. Okay, let's zing on. Someone mentioned therapeutic cloning earlier, so I want to get onto therapeutic cloning. Oh, poor Blue Rook Chess. Thank you, Ishil. Blue Rook Chess got absolutely destroyed there. Never mind, eh? Poor Blue Rook.
Let's take a listen. Oh my God!
Yeah, Blue Rook Chess, we just had to get slapped out the live there. I'm sorry, but you can't be going on like that. So, yes, I will go through culturing microorganisms as well, but let's bust out the stem cells and then we'll take a little break at the end of Unit 1, a little 2-minute break so I can recharge a coffee. And in medicine, stem cells come from bone marrow are already being used to replace faulty blood cells. Okay, yeah, we know that. One day we will be able to use embryonic stem cells to cure paralyzed people, spinal injuries, grow new organs, da-di-da-di-da-di-da-di-da. So you've got the idea. Now we have something called therapeutic cloning, but before I get on to that, yeah, let's do therapeutic cloning first. So we have an issue. Someone mentioned therapeutic cloning, so I just want to bust out. Look, you need to know these concerns. If you don't know these, get to know, make a note of them. Concerns with stem cells. There are ethical concerns because stem cells often come from embryos, and some people say embryos are potential life. So you're taking a potential life. If you took that embryo and placed it in a woman's uterus, a baby could be born. Some people say you've decided to chop the embryo up. Obviously, it can no longer become a baby. But Also, if we put an organ grown from stem cells in someone, remember viruses hide in cells, there could be a virus in the cell. So that is on the AQA Biology syllabus, that some scientists are worried about viruses being in cells. Other things. That'll do. Viruses. All right, let me get on with therapeutic cloning. One of the issues, right, is you guys, your cells. So this is just going to explain therapeutic cloning for you. Your cells have unique markers, right? Your cells have unique markers. If I take an embryo, it's obviously not your embryo, right? We just got spare embryos, mainly from IVF. People who go and have in vitro fertilization at clinics, we get extra embryos around, and we can use those because we might make 20 embryos and they just have 2 kids, and now you're left with 18 embryos. And if they give you permission, you can use those embryos for scientific research, stem cells treatment, and all of that. So we can take an embryo, and I mean, you can just take, as long as someone donates sperm and donates eggs, you can make your own embryos in the lab and put them in the freezer. Now, if I take an embryo and I'm like, oh, I need to give this person a new, what do we want to make? We want to make a new heart for this person, right? We're going to give them a new heart. The problem is that when I grow the new heart, the heart cells will have different markers. Why? Because they don't have the patient's DNA. This is our patient, and they have different DNA. So the protein markers that we call antigens will be different. You can just call them proteins, but the proteins on the cell membrane will be different. So what we can do, we can take the embryo cell, and this is called therapeutic cloning, and we're going to take some DNA from the patient, and we're going to place it in the embryo cell, the stem cell. So this stem cell, we've now placed DNA from the patient. Guess what? It now has the markers. Why does that make a big difference? It means less chance of rejection. So I'm going to type the bit that you need to remember for this. So all you need to say is, oh my good Lord. Therapeutic cloning uses the patient's DNA. Oh my days. Uses the patient's DNA in the stem cell. So the organ grown from the stem cell has less chance of rejection. Bosh. And that is what you've got to say. Come on, Tom, sort it out, you Wally. There you go. Right. Don't overthink it, right? I've gone a bit too detailed there, but it basically means that the organ will have the same protein markers as the rest of the patient's body. So the white blood cells will come past and they'll be like, leave it alone. It's part of our body. It's a self cell rather than a foreign invading cell. Okay, that's called therapeutic cloning. You can say DNA, or, you know, on my bit of writing over here, I've said the word genes, so you can say it contains genes. Genes, DNA. DNA is a gene is a small section of DNA. How can I turn that back? Go on, get off. Oh, you doesn't want to know. Right. Any questions about therapeutic cloning? Okay, did I lean? I may have leaned on my mic. Can you guys hear? You know what? Best to know therapeutic cloning. I think it is on Combine, but it might not be. But, you know, what can I say? I can check it out for you. When I go for the break, I'll check it out for you when I finish cells. Disadvantages of stem cells is going to be that viruses can hide in the stem cells and they could be passed on to the patient. So imagine this stem cell had the HIV. I mean, why would it have HIV? HIV invades white blood cells. But let's imagine there is a virus hiding in it. When I grow the person a new heart and I transplant it, those cells will have the virus hiding in there and the patient will get the virus. Now, it could be a deadly virus. And that human embryos are destroyed, which are potential life. So they're my main two issues. Some people may say stem cells as well. There might be unknown effects or something like that from growing organs from stem cells. One other thing, guys, is that if you just say ethics on some questions, you have to read the question carefully. If it says disadvantages of using stem cells, read the question carefully. If it's just open-ended like that, you can say ethical reasons, but you've got to explain it. It's like saying repeat in an experiment. Don't say repeat. Say repeat and compare the results and see if there's any anomalous results. Remove them and calculate a mean. So when you're writing up an experiment in physics, chemistry and biology and you want to say, oh, they should do repeats, say why. Say so they can compare the results and spot anomalies. So it's the same with ethics. What they got tired of in some of the mark schemes is people just writing ethics. So they want you to say, what's ethical about it? Well, stem cells come from an embryo and that embryo could have been life. You've now shown the examiner you understand what the ethical situation is. They may say ignore just if they put ethics. They have to explain that it's potential life. Right. Plants have stem cells. Unlike humans, they keep them for their whole adult life. I know there's axolotls that have stem cells. They can grow limbs. You can chop bits off of them and they grow them back. Some lizards as well. The tip of the shoot, which is the top of the plant, and the tip of the root, the bottom of the plant, is known as the meristem tissue. This contains stem cells and it's where the new growth happens in plants. And what we can do is chop a little bit off and we can do quick and cheaply make identical plants. Thousands of identical plants. It's quick, it's cheap, and they will all have the same good genes. Unfortunately, it means they're more susceptible to getting wiped out if there's like a change in the environment. But that's not really in this part of the syllabus. So you can, they'll all have disease resistance or whatever. Right, let's move on. Let's go to the cell cycle. I reckon we want to do a bit of cell cyclage. Let's do a bit of cell cycle. Yeah, let's go. Okay, I'm going to get rid of that and I'm going to do some cell cycle. Really simple, really simple. This depends on your exam board. I'm going to smash through this. I say smash through, I'm taking so long today. I'm taking longer than normal. What the arse is going on here? Right, here we go, guys. Gonna be a long one. I will do all the stages of drug testing, but I'm going to do that. That's probably not going to be for an hour. I've got to go through organisation. I haven't even finished the first unit yet. Oh my days, this is disgusting. How long have I been on? How long have we been on? We've been on an hour. Okay, so this is taking longer than I thought. But I think the first two units are going to take just over an hour each. And the second two, bioenergetics, is like 40 minutes. And communicable diseases is going to be, infection and response can be like 40 minutes. So the first two are the longest two. So anywho, enough of my whiffle waffle waffle. Let's look at the cell cycle. So I'm going to go through this for you. If you're asked about the cell cycle, peeps, it happens in 3 parts. So don't get on me about the state of this, because it's a cycle, so who cares, right? Stage 1, it's going to be the biggest stage. Stage 1, is this necessarily stage 1? It doesn't really matter, the order, because it's a cycle, so you could start anywhere, Right, it goes on and on, just keeps going round. Stage 1 is growth, and I'll explain that. You can make notes if you want, and duplicating chromosomes. I guess we can say DNA. I guess we can say chromosomes, or we can say genetic material. Okay, so what happens in stage 1? Growth and duplicating of the DNA. What do I mean by growth? Growth means making more, I'm just giving one example, ribosomes. It's nice to give an example if you get a big fat question. Making more protein, making more ribosomes, making more mitochondria. If you're going to split in two, and I'm going to split in two as a person, I need to double my kidneys because I need 4 kidneys so I can split into 2. I need 4 lungs so I can split into 2. So the cell's the same. It needs to double the ribosomes. It needs to double the mitochondria. So that all happens in this growth stage. Now, stage 2 is mitosis. What happens in mitosis depends on your exam board. AQA is dead easy. For AQA, we just need to say these 2 bits or 3 bits for mitosis. Chromosomes line up on center of cell. In fact, and then I'm going to say chromosomes pulled to each end of cell. Oh my days, I could have done more room. Oh, that's crap, isn't it? Chromosomes pulled, let's just say to opposite ends of cell. Okay, didn't leave myself any room there. So the chromosomes line up in the middle, then they're pulled to the opposite ends of the cell. Then a nucleus forms around those chromosomes. What we don't tell you is that at the start of mitosis, the nucleus just disappears. But in AQA, it's very, very basic what you have to know. Stage 3 is for AQA, cell membrane, and you have to say and cytoplasm divide into 2 identical cells. I didn't leave myself enough room, but you get the idea. That's all you have to know. That's going to smash you all the marks. So stage 1 is for growth, making more protein, making more carbohydrate, making more organelles, so ribosomes, mitochondria, etc. Doubling your DNA and chromosomes. You must say about the duplicating of the chromosomes. That's vital. And then during mitosis, the chromosomes line up on the middle. They attach to things called fibers, and they're pulled to the opposite ends of the cell. And then in cytokinesis, that's not on your syllabus, but they will sometimes accept it if you write it. Cytokinesis is the name of this stage. The cell membrane and the cytoplasm are torn in 2 and you are left with 2 identical cells. Any questiones? The liver, you've got to know that it contains bile, and which it produces bile, doesn't store it, that's the gallbladder, and bile is needed to emulsify fats and neutralize acids. You also need to know that the liver is where lactic acid is broken down. No, Ashir depends what. Yes, no, not really. You've got to know what happens in them. So growth and doubling of DNA in the stage 1, you call it the growth stage. The second stage, mitosis. If you're doing AQA, that's all you've got to know. You'll get all of the marks. You don't need to know about prophase, metaphase, anaphase, telophase, spindle fibers and that business, not for AQA. The rate I'm going, I would be surprised if I'm still going when your exam starts. No, you don't need to know the life cycle of a mosquito if you're doing AQA biology. The only thing you need to know about a mosquito is that the mosquito acts as a vector and it carries the malaria protist. The malaria protist is actually called the plasmodium, and it does not affect the mosquito. The mosquito is not harmed by it, so it lives inside the mosquito, and when the mosquito feeds, it swims down its proboscis into the bloodstream of the person the mosquito's biting. The mosquito's not trying to do the malaria a favour, the Plasmodium protist a favour. It's trying to get glucose from the blood of a human so it can do respiration. It's feeding on us. And it just so happens that it unknowingly transfers the Plasmodium into us. Now, you do need to know 3 ways to prevent that. I'm going to go into communicable diseases later. We are going to do communicable diseases. But I'm going to do the heart first, and I'm going to do... You can say identical daughter cells or identical new cells. Yeah, it's good to say new cells or daughter cells. Not in paper 1, you don't need to know about the kidneys. Not in paper 1, Nell Rain. That will be paper 2. You don't need to know a lot about them either. A protist and a vector. So, okay. A vector is in biology. Remember, a vector is something different in physics. So a vector in biology is an organism, a living thing that can transfer something else. So it's used as a transfer mechanism. So the mosquito is the vector, not the protist. The mosquito, or the insect that carries the protist, the malaria protist, is the vector, not the actual disease-causing pathogen. The pathogen's not the vector. The vector is not harmed by the pathogen, but it passes it on to another organism. And that is what the vector is. Protease, yeah, that breaks down protein. Let me make a, you know what? Give me 2 secondos. Bada bada bing, bada bada bong. Let's make a couple more admins. Moderators. There you go. I've got another moderator there. Ta-da! That means for the live sessions you can do a bit of moderatione because, you know, by the way, don't feel pressured that you got to like moderate. Oh, that was some questions I did on stem cells and stuff. Right, let's just smash a couple of questions out on cell cycle. Let me smash out a couple of questions on the old cell cycle. Let's do a couple of cell cycle questions. I need to get off of cells, man. I'm going to end up doing the whole live on cells. This is just taking far too long. I need to speed up. Let me speed up. Right, I'm going to race through because it's gotten too long. Right, before a cell begins to divide, its DNA replicates to form 2 copies of each chromosome. Describe one other change that occurs in a cell before the cell begins to divide. Cancer is caused by changes in cells that result in uncontrolled cell division. Before a cell begins to divide, its DNA replicates to form 2 copies of each chromosome. Describe one other change that occurs before the cell divides. So other than DNA or chromosomes doubling, what else happens? Grows larger. Nice, I like that. It gets bigger. More ribosomes. Okay, should I write more ribosomes? I should say it makes more ribosomes. Let's say makes more ribosomes. Makes more organelles. Listen, you can call them cell structures or subcellular structures. So those are some things you can go through. Makes more protein. Name structure Z. What is Z, do you think? Ooh. Anyone know what structure Z? Thank you, Isha. It's the cell membrane. Watch out for these, man. They'll try and trick you up. These are called fibers, by the way, just in case you wanted to know. They're actually called spindle fibers. Why, you're never going to get asked about. That's known as the centriole. That's not part of your syllabus. These are chromosomes being pulled apart. Hornet moth. The cells which are first formed by the fertilized eggs of the hornet moth are stem cells. Name the process. What do we call the process by which stem cells, Lucas differentiation, when they become specialized, we call it differentiation. Lovely jubbly. Right. This question is about cell division. I'm just going to do a couple of these, then let's move on because we'll be here all night. We'll be here all night. This question's about cell division. Which process makes 2 identical new body cells for growth and repair? mitosis, smashed it. And then stage of the cell cycle, stage 1, stage 2, stage 3. Look, AQA, they ain't even saying like prophase, anaphase, telophase. You don't need to know any of that. So, let's go. Cell grows and chromosomes replicate. You see, I wasn't lying. One set of chromosomes pulled to each end of the cell. Stage 3, cytoplasm. Notice we need to say and membrane divide to form new cells or daughter cells. Boom! Okay, actually, that's a good point. So all I can say is that specialization is like the end of the journey. Differentiation is the journey. So you are a stem cell, and differentiation is the process by which genes in your DNA are switched off. You don't need to know this. That's A level. Certain genes are switched on and off by various mechanisms. That's called differentiation. And then you become a specialized cell. So the journey is differentiation. And at the end of it, we say you have gained specialization. Specialization is what you are at the end. You have gained specialization. Your specialization is to swim to an egg and fertilize it, or is to absorb water. So specialization is what you gain. Differentiation is going from a stem cell to a specialized cell. Okay, last one. What? Here we go. Come on. I need to move on. Genetic material is made of many small sections. Each section codes for a specific protein. What is the one section of genetic material on a chromosome called? What's one bit of a chromosome called? I can't say I'm better than anyone, Lucas. You have to make that your mind up yourself. You go, you have to make your mind up yourself. And describe what happens during each stage of the cell cycle. So we already did this. So this is worth 4 fat marks. AQA, can you say what happens during each stage of the cell cycle? Bash through it. We've already done that. The question is about the cell cycle. Chromosomes are found, blah, 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 are copied during the cell cycle. Tick one box. Where are chromosomes found? That's dead easy peasy, isn't it? What is the name of a section of a chromosome that controls a characteristic? That's a gene. And then over here, we're just going to say growth. DNA replicates. Replicates is another way of saying duplicates. I'm going to bush over here to stage 2 and say mitosis, and then I'm going to add chromosomes pulled, oh my gosh, to opposite ends of cell. Remember, I kind of need 4 things, so I'm trying to say 2 things about each stage to kind of make 6 things. Stage 3 is cell membrane and cytoplasm divide. Now, if I had room, I would write to make 2 identical daughter cells or new cells. Bish, bash, bosh. Which cell is not dividing by mitosis? Which cell here is not dividing by mitosis? The nucleus. Okay, the nucleus doesn't split into 2. No. Let me clear that up for you now. So what happens? Oh my gosh. The nucleus does not divide into 2. So what happens actually is, here is your cell beforehand. The nucleus. Let's draw the nucleus. Here's your chromosomes. The nucleus disappears. Ta-da! That's actually in the first stage of, at the start of mitosis, the nucleus goes. So we just have cell membrane, cytoplasm, and the chromosomes. And then what happens is they line up on the middle like that. Then they pull to each end. Then they bubble up like this. And then what happens is 2 new nucleuses reform. So at no point is the nucleus torn into 2, only the cell membrane and the cytoplasm. So 2 nucleuses reform like that, and then the cytoplasm and cell membrane tear into 2, but the tearing is after mitosis. Right, the cell that is not dividing is C. See, look, you can't see the chromosomes, as you can see the wiggly chromosomes there. Explain why the student used the tip of the root. it contains meristem tissue. And remember, the meristem tissue, oh my gosh, dividing. It's got a high rate of mitosis. They squashed the tip. The slide was placed on a stage. Microscope. Describe how to use the microscope. Oh, we've already done that. I'm not doing that. Describe how to make the data blum blum blum. Right, I think I'm going to get off of this. I'm just taking far too long on this. Let's move. Let's move. Let's go. Let's go. Yeah, let's go. Let's go. Let's go. Let's not do these microscope. I've got millions of these questions. Let's just zing. I can always come back if there's some people that want, yeah, blah, blah, blah. What structure is Z? That's a chromosome. Mitosis is important to repair tissues. What is the function of the nucleus? Contains DNA. Name one type of cell that, yeah, we know that. Plant cell. What is this? Oh my gosh. All right, 6 marker peeps. Oh, let's move on. Let's move on. Let's move on. Let's not do the 6 marker on that. Let's move on. I'll come back to 6 markers later. I'm going to move on. I'm going to move. On. Oh, let's go here. Right. Let's go here first of all. Transport in cells. Oh, please get off of cells. Guys, it's like 5 more minutes. Then I'm going to take a 5-minute break. I'm just going to do transport in cells. Then the osmosis core practical. Then we're done with the first topic. Unless anybody can think of something I haven't gone over. So particles move into and out of cells by different processes. What you need to do is just remind yourself for tomorrow that you know how water would move into a root hair cell. So can you say 2 things about how water moves into a root hair cell? Bish, bash, boshicus. Can you explain how phosphate ions would move in this diagram? Don't just say, oh, it's an ion. They always move in by this. Use the diagram. And then you can see how would the magnesium ions move in. So it's really 2 marks, 2 marks, 2 marks. Water is osmosis from a high to low concentration. Make sure you write the full word concentration. Make sure you say high concentration outside the cell to a low concentration inside the cell in this situation. Phosphate ions. How would a phosphate ion move in, peeps? Yes, Lucas, it is by diffusion from a high concentration to a low concentration. That is a passive process, does not require energy. I would include that. Osmosis is passive as well. And then magnesium ions are going against the concentration gradient. That is going to be active transport, requires energy from respiration. Boom. And that should get us all 6 marks, peeps. Nice. Lucas, there are 2 ways to say osmosis. I'm glad you brought that up. So you can say water moves in from a high water concentration to a low water concentration, or the more common way in the exam board, when we go to the osmosis practical, we'll look at that, is to say that water moves from more dilute. Now, if you think about dilute, that would mean I've got almost pure water. So that's the same as saying high water. So you could say more dilute concentration to a higher concentration. And that messes with people's mind. The moment you're talking about dilute to higher concentration, you're talking about the salts and minerals. So you're saying it's in a more dilute and it moves to more concentrated. Or you say it moves from a high water concentration to a low water concentration. Keep one of them, stick with one of them. Bish, bash, bosh. Okay, here, no, this is not triple. This is for everybody. Everybody. There's your marks. So you can check your marks. Water is always osmosis. This is a 6er. Study with Vanny. This is 6. Root hair, Michelle Emmanuel Y5T. Root hair cell does active transport for most mineral ions. The reason why is not that mineral ions have to be transported by active transport. It's that the soil naturally on the earth, anywhere we go on the earth, the soil is lower in minerals generally than the plant already has in it, and the plant still needs more minerals. Okay, so the resource around plants is low in minerals, so it has to use active transport. If I happen to put loads of phosphates in my garden, then it's going to diffuse in because it will go from high to low. So it's just so happens that it is mainly active transport for minerals. You can see the marks there. Okay, yeah, let me know if there's someone you want me to kick, but I got some people that can do some kicking. I'll get to the triple dipple. The triple is going to be really for cells. This first topic, the only triple is the agar. You know the one I'm on about, the culture, microorganisms. Yeah, I'll get to that. Let's do this. Microorganisms break. Okay, a student investigated the effect of different concentrations of sugar solution on pieces of carrot. By the way, they can use potato, carrot, turnip, any vegetable you like, maggot, worm. They could use anything. They used eggs one year, so eggs without the eggshell. So you're still going to get osmosis. So all we have to do here, I'm just going to whiz through this. I'm not going to take long over this at all. They calculate the percentage change in mass. Why do we calculate percentage change in mass? The idea is they have different starting masses. This is why we use percentage change. So easier to compare. So what we're saying is, if I have a 10 gram potato and a 5 gram potato, and the 10 gram loses 1 gram of water and the 5 gram loses 1 gram of water, the 5 grams lost 20% of its weight. The 10 gram has only lost 10% of its weight. So it looks like they've lost the same amount, but relative to their mass, they have lost different amounts. It's better to use percentage because they might all have different starting weights. So, couple of things they could have improved this experiment. They could have put lids on to stop evaporation of water, because that will change the concentration. So I might think I've got 0.2, but if I get evaporation, it will change my concentration. So best to put cling film or lids over this. The other question they can ask is, when you take the potato out to reweigh it, and you reweigh the potato, you should dry it first. That's supposed to be a cloth, by the way. Get a paper towel or a cloth and dry it first. The reason is that it will have water stuck to it called excess water. Now, this potato from here might have more water than this potato from here. In which case, if I weigh them, it's not fair because I'm not really finding out how much the potato changed. So, put lids on, dry them. You've got to leave it for 24 hours. You've got to give osmosis time to happen. Okay. Try to keep the surface area the same. Otherwise, that will affect the rate of osmosis as well. And then what you're going to do from this, you're going to plot these bad boys on a graph. So you're going to get a graph of percentage change in mass over here. I'm going to do this nice and quick for you, man and girl and people. Whatever. I'm not even going to write percentage change. You get the idea. That is percentage change. 24, oh my gosh, 5, 10, 15, 20, 25, and then down the bottom we're going to go down to minus 15, so minus 5, minus 10, minus 15, and then 24. So at 0 it was 24. Let's just bash this in. Don't get upset if I haven't put it in the right thing. What is that? That's 10. Oh my gosh. 24's there, so 24 is there. Then I want to go 0.2 and 12 there. Roughly. Don't beat me up for this because I'm trying to speed. And then that is 1% change in mass there. Then I got 0.6 is minus 8. Oh gosh. Let's go there. And 0.8 is minus 15. Bosh. Right. Now you guys can see, excuse me for not using a ruler. Oh, actually, let's just straight line that. Right. Okay. You get the idea. Look, what they're going to say is estimate the concentration of sugar concentration inside the carrot cells. How would I find what the actual concentration of sugar is in the carrots? Thank you. When the percent, good. When this is 0, so we're going to go here. We're going to zing there. I reckon that's 0.43. 0.43. That is the concentration. Where percentage change is 0, what that tells you is that it doesn't mean no water has moved in. What it means is the same amount of water has moved out by osmosis as water has moved in. It's dynamic. There's movement here. So water's going in at the same rate as out, so it doesn't change size. That means that the original concentration of the cells inside the carrot was 0.43. Now, what I'm going to get onto this and the old dilute, explain why the mass of the carrot at 0.6 went down. So why did my carrot was like that, and then it shrunk like that. So why did it go down? Why did my carrot go down? This is how I am going to argue it, and then I will tell you how you can argue it. So if I'm told to do this, I'm just going to say water moved out. It's 4 marks. Water moved out. I'm going to say by osmosis. That's possibly 2 marks smashed right there, right? Water moved out by osmosis. Now you're allowed to use water concentration. So from a high, oh my days, water conc inside carrot to low, oh my gosh, this is disgusting, isn't it? To low water conc outside. Notice I've used the location. I've said the water, I should really say cells, the high water concentration inside the cells to a low water concentration outside the cells. Gosh, and that's going to get me 4 marks. I know I put 3 bullet points. I'm going to put across, because it's 4 marks, across a semi-permeable membrane. A better one is partially permeable membrane, but it's just too long for me to write. Cool. That's going to get me 4 fat marks. Now, I know what some people are saying. Sir, do you have to say water concentration? The other way to word this, so check this. I'm going to change the wording of this bit, right? And this is the way I word it. But in the exam board, this will be in the allow section. In the left-hand corner of the mark scheme, it will be written like this. So if you're the type of person that's like, nah, I don't want to do the cut stuff that's in the allow, then, which doesn't matter because, or accept, the accept is totally scientifically correct. I find that easier. This is the alternative way. Water moved out by osmosis from a more dilute solution to a more concentrated solution. And there you go. So if you prefer that, bust that out in the exam. Dilute is the same as saying high water, right? If you're dilute, you have lots of water, so you're high water. If you're concentrated, they're not talking about water concentration, they're talking about salt, sugar concentration outside the carrot, which is low water. So I tend to go from high water to low water just because I remember it like that. But the exam board often talk about water moving from a dilute to a more concentrated solution. So you choose. By the way, instead of saying that, you can say down a concentration gradient, you know, but I don't know, best not to with that. Yeah, anyway, let's move on. Last thing, and then it's break time, okay? Because we have completed the first unit. I know I've taken far too long. I will speed up. I promise, okay? Last thing before we move on. You can say partially permeable membrane. Osmosis only happens across a partially permeable membrane or semi-permeable membrane. The better word is partially permeable. It only ever happens. Like if I spill water on my table and water moves on the table, that's not osmosis. It has to be going across between cells, in between cells, and then it is partially permeable. If it's crossing a cell, then it's osmosis. Sorry, what am I waffling about? Right, listen, peeps. When we do the bacteria practical, this is triple only. I'm going to do this for 5 minutes, then I'm moving on. So, Listen, have I got one of these so I don't have to do one of my bent drawings? Let me have a quick flick up on my slide over here. I might have a little thing up here. Let's go, let's go. Come on, Tom, you dweeb. Yeah, there we go. Bish, bash, bosh. Okay, we'll deal with it with this question. Let's see if you guys can do this question. A student is given a tube containing a liquid nutrient medium, contains one type of bacteria. So I'm reading these. It's got one type of bacteria, nutrients. You can think about what nutrients they would be if you ever get asked that. The student is told to grow some bacteria on agar jelly. Remember the nutrients, you don't need that for this question, but they would contain glucose, amino acids, fatty acids, glycerol, lipids, protein, starch, glycogen, whatever. You get the idea. Vitamins, minerals, things that any living thing needs to be healthy. The student is told to grow some bacteria on agar jelly in a petri dish. Describe how the student should prepare an uncontaminated culture of the bacterium in the petri dish. Basically, how are they going to make this dish? You don't need to talk about antibiotics. How are they going to make a dish of bacteria which will look like that? It'll look like this. So here's my petri dish of bacteria. So 6 marks. See if you can explain how they should prepare a petri dish of bacteria. Now, dilute is a high water concentration, but you can say dilute as in a low concentration of salt. So you can say water moves from a low concentration to a more concentrated solution, and you'll get the marks for that. So you can say that. But if you're talking about water concentration, what they're talking about is salt or sugar. We call them solutes. They dissolve in water, and they make, so you can talk about water moving from a low concentration to a high concentration, but that can mess people up and make them think it's active transport and requires energy. It's not, it's passive, because the water is moving from a high water concentration to a low water concentration. Or you can say a low concentration to a more concentrated solution or high concentration, but then you're talking about salts or sugars or solute, whatever your solute is. Okay, cool. Oh yeah, you got some nice points here. So what you want to do is sterilize your inoculating loop. I'll put the answers up in 2 seconds so you can check. But so you're going to want to sterilize your inoculating loop. That's the metal thing. You're going to dip that. They've already told you you've got the medium with one type of bacteria. So you want to get a petri dish, agar jelly. How long should you take the lid off for? If you take the lid off fully, bacteria from the air that aren't your one are going to get in there. You only want your one. So you only lift the lid slightly. Work with the lid just slightly open and for the least time possible. Sterilize the inoculating loop in a Bunsen burner. Dip it in the broth. Zigzag it without breaking the agar jelly across the agar jelly, and then quickly close the lid. That's obviously important too. Now, zigzagging just helps the spread of it evenly across the agar jelly. Couple of other things. Someone said in the chat, really good point, work close to a Bunsen burner. What it does is it creates a convection current which blows away any of the bacteria and microorganisms in the air that you don't want getting in there. You can say sterilize the agar jelly. What we mean by that is you can like warm it and sterilize it by killing any bacteria that are in there. Are we saying we're in a classroom? If we're saying we're in a classroom, you might consider saying don't let it go above, and then you want to seal it, inoculate it. You want to seal it and incubate. Incubate means put it somewhere for it to grow, and you don't want it to go above 25 degrees if you're in a science classroom. If you're in industry, you can use it at 37 degrees. The reason why we can't let it go above 25 degrees is if we do it at 37 degrees, we'll get harmful pathogens that love to live in the human body. You need to tell the examiner, I know the human body temperature is about 37 degrees. So don't do it at a temperature where harmful bacteria will grow. Why do they grow at that temperature? Because they're the ones that like to live in humans and hurt us. So in industry, we do it at 37 or higher. We do it at higher temps because we get a faster rate of reaction. Anywho, this is the mark scheme, so you guys can see the mark scheme. Oh gosh, I've noticed you keep slapping people out the chat. Well, you know, if he got slapped, he got slapped.
Let's take a listen. Oh my God!
Well done, well done. Sandeep got the licks. Yeah, good if you got that. Now, why are... Okay, let's look at this question. Let me make this question a little bit bigger. And then we're going to go on a little coffee break. And then we'll come back and we'll get into organization. Once I've broken the back of organization, it'll be a lot quicker. Oh my gosh. Yes. So after the culture has been prepared, the student added 1 drop of each of 5 disinfectants A, B, C, D, and E onto the culture. The diagram shows the appearance of the Petri dish 3 days later. Why are there areas of agar jelly where no bacteria growing? What happened to the bacteria? It killed them. Yes. Yes, this is triple. Triple higher. Yes, it's been killed by... Is it disinfectant or antibiotic? I think it's disinfectant. I think you're right. Yeah, it's disinfectants. Well done. Disinfectants are different than antibiotics, so watch out for that. Antibiotic is a drug that kills bacteria. Disinfectant also kills bacteria. Or we can just say the bacteria cannot grow there. They can't reproduce there. So we can say it is where the bacteria cannot reproduce or where they can't grow, or I would just put that it's where the bacteria are killed. Okay. And then the student concluded that disinfectant D is best for using around the home. What is a reason why D might not be best? It's the best at killing the bacteria, but why might not it be best for putting in your house? Yeah, they would accept work close to a Bunsen burner. They would accept work close to a Bunsen burner. That's like an A-level thing. They will include those. The examiners have got A levels in the subject. They will include those. Yes, I think maybe toxic to humans, side effects. We'll have a look at the mark scheme, but I would say that it might cause side effects, something like that. Maybe too strong. Well, you probably can't say too strong. It might be harmful. Might cause reactions with the skin or something. I don't know. Give one reason why the student might not... Oh, yeah. One reason why they might be correct is it killed the most bacteria. One reason why they might not be correct. Let's check the mark scheme on that one. See what they're saying. They've got bacteria killed or destroyed for the first one. Then they've got largest area where no bacteria are growing. D may be harmful to people, animals, surfaces. Yeah, may work differently with oh yeah, this was only done on one type of bacteria. The other thing, if they say something like, would this be a good one to evaluate whether this is a good antibiotic, antiseptic, disinfectant, whatever the practical is about. If they say evaluate whether this would be a good one to use, just say, well, they only did it on one bacteria. There are thousands of species. We don't know how it will affect other pathogens or microorganisms, fungi, things like that. So, yeah, always think about that too. Right. Let's take a little break. All right, we're going to take a quick 2-minute break, guys. Then when we come back, we'll do organization. Yeah, I think so. Too expensive, maybe price. They didn't say price. Oh my gosh. No, it's not triple only. We're going to be heading back to the combined bit after this. So we did binary fission, which is triple only, and this bit is triple only. Guys, taking a quick break. I will be back in 2 minutos. Ay, I am back. Okay. Oh my days. Let's go. Alright people, let's get on and make sure you have hit the like. That will massively help out the channel.
If you push the button,
Like me.
Would you like it?
Like.
do it. Do it.
Do it.
Yeah, I haven't had my coffee yet. It's here in my Mickey Mouse club, like my Mickey Mouse teaching. Yeah. I haven't had it yet. This one's crusty. It's instant. It was only a quick 2-minute break. So, let's move on. What am I doing here? What are we doing here? We're going on to organize. I've got hundreds of questions on organization. Jeez, and all of this. Let's just move on. Where am I? What's going on? What the? Jeez. Yeah, we're going to go through. Oh my gosh. Oh yeah, you can see if you can label this. You can put it up in the chat if you can remember what all of this is. And then I'll just crack through for you guys. And then, yes, we're going to cover everything. So I will do the lungs. I will do all of the business. So, if you know how to label that, don't bother putting that up in the chat. If you think, yeah, I've got all of that under control, then all you need to do is lick over onto here and 3 marks to 4 marks on how protein is digested. So if I said a rat eats a snail has protein in it, how is the snail digested in the rat's stomach? Bosh, that's putting it in context. So you might be like, oh, I don't know, it's an animal. So it's the same way that we digest protein. What role does bile play in digestion? Bosh, could be 4 or 5 marks right there. So, yeah, I like some of these answers. So I'm going to put you the 4 marks down right here. Let's do it. Bish, bash, bosh, bosh, bosh. Excuse the writing, guys. How is protein digested in the stomach? A lot of you have put this up, so well done, yeah. Come on, Tom. Sort out your pen. Sort out your board. That's what happens. Yeah, some wicked stuff there. So, protein is digested in the stomach. So, protease, which is the enzyme, you can say pepsin. Protease breaks protein into amino acids, and you'll smash some marks for that. HCl, now I'm not writing hydrochloric acid, but you could. Hydrochloric acid creates pH 2, optimum for protease enzyme or pepsin. To be honest, I should really say pepsin. Hey, what's the side role that hydrochloric acid also plays? It also plays a role not to do with digestion. Does anyone know what the hydrochloric acid does not to do with digestion? You guys have got some wicked answers here, so hopefully you'll get some of these coming up. Anybody know? Do I have to say pepsin? Yeah. No, sometimes. Now, I know that's going to annoy you, but technically, there are some protease enzymes that don't like a pH of 2. So I would say you can say protease breaks protein into amino acids. They will give you the marks for that. And you can say pH 2 is the optimum conditions for this enzyme. But it's that specific enzyme that's pepsin that loves the pH of 2. There are other protease enzymes that like pH 3, pH 4, pH 1. So specifically, actually none like pH 1, but yeah. You get the idea. So they could get all, you said protease likes pH 2. They could get all arsey like that. But generally, if you're just saying that pepsin breaks down the protein in the stomach to amino acids, you could also just say protease there. But it's better to say pepsin. Right. What role does bile play in digestion? It has 2 roles. By the way, hydrochloric acid also kills bacteria, but that's not really to do with protein digestion. So you wouldn't pick up any marks for that, but you wouldn't lose any marks for putting that in there. What role does bile play in digestion? We can say bile emulsifies fats. Now, lipids, yeah, you can say lipids, same thing. Bile emulsifies fats or lipids, makes smaller droplets. Basically, fats turn into droplets because they repel water. You ever got oil on your hands? It's hard to wash them off. So they form droplets when you eat fat in your digestive system. This just makes them into smaller pieces. It's like you chewing up a piece of bread. The bile makes the fats into smaller droplets, means larger. You should write it, but surface area. You don't need to say surface area to volume ratio. You can just say larger surface area. So lipase, that's going to be a mark, enzyme, can break down the lipids more rapidly. Now, I would probably go on to mention into fatty acids and glycerol. Now, yes, in your exams, you're going to miss something out. Be prepared tomorrow that you will miss some stuff out. That's not the point. Everybody forgets and comes out and goes, oh, why didn't I put that? That's why you don't have to get 100%, because they know you're going to forget some stuff. It's about trying to go through your head and be like, bile, this is how, and know your stuff. Right, bile also, second job, it neutralizes, oh my gosh, neutralizes stomach acid. Now, the stomach acid is HCl, so we can say it neutralizes stomach acid. Why? And that's just to create the perfect conditions for amylase and lipase in the small intestine, to create alkaline conditions for amylase and lipase. Essentially, do you have to go to that extra? I don't think so. That's a whole heap of marks right there. Right, any questions? Any questions? Any questions? Does everyone know where bile's made and where it's stored? It's made in B, which is the liver. It's stored in G, which is the gallbladder. E is the large intestine for absorbing water. D is the small intestine for absorbing small food molecules like glucose, amino acids, fatty acids, glycerol. C is the stomach, which contains the hydrochloric acid and is where protein is digested, which we've gone through there. H is the esophagus. Not a lot to remember there other than that it transfers food to your digestive system. If you did GCSE Edexcel Biology, you would need to know about peristalsis, contraction of the smooth muscle that moves the food down, but you don't. A is the salivary glands. It's the mouth really, but the salivary glands are back here. They produce the enzyme amylase. Now, there are two organs that produce all three enzymes. Who knows which organs produce? Two organs in the digestive system produce all three enzymes, as in protease, amylase, and lipase. What are the two organs? Small intestine and pancreas. Check that out. Boom! Well done. Yes. Stomach only produces protease, and it's in the epithelial cells, which is the lining of the inside of organs. So there are epithelial cells in the small intestine, large intestine, the stomach. They're the cells that line organs on the inside. Epidermis is on the outside. Epithelial are on the inside. Okay, what are we doing? We need to do the food tests. So I'm going to do some quickly zing the food tests with you guys. They often do 6 markers on this. So, oh my gosh. That is not what we want. There we go. Hey! Let's smash out the food test quickly. So, what is the test for sugar? Yeah, I will go through Villi. And Villi. Excellente. So, the sugar test is Benedict's. Now, you can say reagent. And Benedict's reagent. Oh my God, it can't be 8 o'clock already. It is. What a dipstick. Benedict, I'm going to speed, he says. He's not really. Benedict's reagent, warm it. Yes, people can say heat it for 70 degrees for 5 minutes, whatever. I'm just going to put warm it. Turns blue to red. And PPT, it's a solid, a precipitate. Okay, that's potentially 3 marks. Okay, not often, but Benedict's reagent, make sure you heat it. It will go blue, form a red solid precipitate. Yes, it can go yellow, green, etc., but that is when there's less sugar. Protein is biuretes. Hopefully you got that. You can call it reagent or solution, same as that one. And the color changes blue to purple. And you can remember that really easily if you're someone who struggles with this. You can remember that by thinking protein begins with a P and purple begins with a P. Protein goes purple. And then you can try and remember the biuretes all by yourself. But that should give you the protein and purple. You can say violet, you can say lilac. There are loads of pink. You can say all of those colors are accepted. Starch is iodine. And it goes orange or brown. Either of those is acceptable, and it will turn blue or black, and it is with starch. Now, lipids they don't really ever ask lipids, but just in case they ask lipids, some people say, oh, you've got to say Sudan 3 test. Okay, so the Sudan 3 test forms a red layer that floats at the top of test tube. Okay, you could say use ethanol, shake a lipid in ethanol, pour it in cold water, it goes cloudy. That's another one. By the way, a quick note, do remember carbohydrates are not just starch. There are simple carbohydrates, which are sugars, and complex carbohydrates, polymers, which are starch. So if you're asked 4 marks, explain how to do carbohydrate tests, you need to do the sugar test and the starch test with the iodine, okay? They do accept Sudan 3 for AQA, red layer, and they do accept emulsion. But what I'm saying is, in the specification, they made a little mistake. So if you read the specification on food tests, it says students need to know about food tests for sugar, protein, starch, and lipids. It then goes on to say specifically students should know how to carry out the Benedict's test, how to carry out the biuretes test, and how to carry out the iodine test. It makes no mention of Sudan 3 or the ethanol, so they have steered away from asking it. You go through all the past papers, you will see it come up on multiple choice, but it's not the selected answer. It very rarely have they come up with it unless they've told you about it, and I think it's because they specifically didn't say students need to know that test. So there's no mention of Sudan 3 or the ethanol test in the biology specification for AQA for combined and trips. So I'm like, have it there, but don't memorize it as much as these bad boys, because you need to know them. Right, Villi. Yes, we go through the Villi. Okay, guys, look at this. Level 3 artwork. I know, amazing. Okay, do I have to draw that all the way around? I can't be bothered, guys. This is called a villus. These little things, we call it the brush border, but you don't need to know that. These little things are called microvilli. They will get you 2 separate marks. Villi, microvilli. They're 2 separate structures. Both increase surface area, okay? Both increase the surface area. How does that help, having a large surface area? For rapid diffusion, okay? Rapid diffusion of whatever they're talking about. Maybe they're talking about glucose. Maybe they're talking about how do amino acids get absorbed. So, what do you need to know? Villi have a large surface area because they're folded. Microvilli is further extra folding, and that means you get a large surface area for rapid diffusion. Now, inside is something that looks like this. This is a blood supply. This is a set of blood capillaries. So, capillary network. you can say large blood supply. I mean extensive. There's loads of words we could use. Large blood supply. Okay, how does that help? That is a description. By the way, that is a D for description. D for description. E for explanation. So check what they're asking you. Are they asking for a description? If they want the explanation that goes with this description, then the explanation is maintains a steep concentration gradient. So that's not what a large surface area does. Maintains a steep concentration gradient is for the large blood supply. That's how a large blood supply helps with absorption in the small intestine. Now, for the next one, I know what you're saying. There's a thin wall. There's two places where there's thin walls. You can talk about a thin wall on the capillary, or you can talk about a thin wall on the villi. I'm not drawing all the microvilli. You get the idea, right? So we can talk about thin walls here on the microvilli or thin walls on the capillary. And so I'm just going to go description. Oh, let's include it. Thin wall on villus, on villi. Thin wall on capillary. Actually, you probably won't get a mark if you say thin wall on microvilli. Microvilli is a single cell, right? So microvilli, they're the yeah, whatever. The villus is not a cell. The villus is a structure. It is a tissue. And so it has thin walls. But we can't really say a cell has thin walls because a cell only has a cell membrane in animals. So be aware that don't get funky and say thin cell wall. Don't say thin cell wall. You won't get the mark because animals don't have cell walls. So thin wall on the villi, or thin wall on the capillary, or thin wall on the small of the epithelial lining of the small intestine. Anyway, enough of that business. The explanation is short. Diffusion distance. Okay, and so you can see how you could get 6 marks. 3 descriptions, 3 explanations. Okay. Yes, you can say it's 1 cell thick. The villi are 1 cell thick, or the capillaries walls are 1 cell thick. Yes, you can access this tomorrow. Just going to quickly go through now. In AQA, you don't need to know the breathing mechanism. Okay. And by the way, any questions, just put them up if you have a question. I haven't read the chat for a bit because I've been too busy waffling. Yes, Michelle Emmanuel, villi and alveoli have the same adaptation. So both have thin walls. So the explanation's identical. So thin walls, short diffusion distance, description, explanation. Now we want to talk about the large surface area. We want to say there are millions of villi create a large surface area and lots of microvilli create a large surface area. If we're doing alveoli, we just say millions of alveoli with a folded surface creates a large surface area for rapid diffusion. This time we're talking about oxygen with the alveoli and carbon dioxide. In the small intestine, we're not talking about gases. We're talking about glucose and amino acids. But the same structural explanations are the same, and both have a large blood supply capillary network to maintain the concentration gradient, because you want a steep gradient so diffusion happens quickly. Awesome. Yes, yes, yes. Trachea is A, B is bronchi, C is bronchioles, and D is alveoli. You've got a diaphragm, you've got ribs, you've got intercostal muscles in between, and pleural membranes. Around the outside you have a liquid and pleural membranes. Okay. What do we want to do? We want to... Oh, no, let's have a look at the alveoli. So, alveoli, look. Alveoli are 1 cell. The wall, this is the wall. It's 1 cell thick. Look at the wall of the capillary. It's 1 cell thick. So, just like the villi, it's short diffusion distance. Now, if it didn't have the blood is moving off and keeping the oxygen flowing. So there's always a low concentration of oxygen in the blood around the alveoli and a high concentration there. We call that maintaining the concentration gradient. Anywho, does anyone have any questions about any of this? What is the names of Mose Wallace? Explain as 6 marker, please. What for the alveoli? Lucas, what do you want for the alveoli or... So a typical question on this is explain how the lung... Okay, so there's 2 slight variations. Slight variations on this. Okay, so we could have slight variations, guys. We could, and there will be a slight variation in the answer. If you put the same answer, you'd basically get like 5 out of 6. Okay, so you could have describe and explain. I'm not going to write the question, it'd be too hard. Describe and explain how lungs, oh gosh, let's just say alveoli are, I don't want to write it out, adapted for, oh, it's just so long, man. We can't do it. Okay, so that's just too long. I should type it, but there is a variation. I'm just going to talk it. Right, there is a variation. So you could have describe and explain how the lungs are adapted. You could have describe and explain how the alveoli are adapted. Now, alveoli, you've got your description. So sometimes it might just be a 3-mark description. You don't need the explanation. So here's the D's. We're going to go thin walls, right? We're going to go lots of alveoli means large SA. And then we're going to say, these are descriptions, that's why I put a D. We're going to say lots of capillaries. I would prefer to say a large network of blood vessels or something, but I'm just going lots of capillaries. Now, that would be my description. You can also, with the alveoli, get away with saying they are moist. That's beyond the specification, but you can say they are moist. Okay, gases dissolve better in when the surface is moist. It creates the contact for like, you don't need to know all that crap. Right, so thin walls, lots of alveoli, large surface area, and lots of capillaries, blood vessels, whatever. Now the explanations are exactly the same as with the villi. So your explanations would be the same. So short diffusion pathway, that goes with thin walls. Lots of alveoli, large surface area for rapid diffusion. So it's identical. And then the lots of capillaries maintains the concentration gradient. Now, making sure, and if you wanted a detail on that, you'd say ensures that oxygen is always higher in the alveoli than it is in the blood, keeps diffusion going from the alveoli to the blood. Yes, this is AQA. Yes, this is AQA. No, I haven't gone over the heart. I'm going to go over the heart. Okay, now let me show you with the lungs. Let's pretend it's the lungs. They say, how are the lungs adapted for gas exchange? They haven't said alveoli, but they've said the lungs. So, I'm going to include all of that. That's going in my 6-mark answer if they ask me how the lungs are adapted. And to be fair, you can smash that out and get all 6 marks. Now, there's controversy over this next bit. I will show you it just to make you feel happy. How does ventilation, so you do not need to know in the AQA specification about how we ventilate the lungs, but if you would like me to, I will go through it, okay? Ventilation of lungs, basically, you can just know that breathing, ventilation by the way is a fancy name for breathing, ventilation of lungs maintains the concentration gradient. Maintains concentration gradient. So we're going to have high O2 in alveoli, low O2 in blood. Right, so the idea is with ventilating the lungs, we're constantly bringing in a fresh supply. You do not need to know the mechanism of breathing. I'll prove it in 2 seconds for you. But if you want me to go through, I will go through the mechanism. Let me show you the mechanism. So the mechanism is, let's say we're going to breathe out. So when we want to clear our lungs of carbon dioxide, right, we're going to breathe out, right? For breathing out, we want to say that oh, let's do breathing in, because breathing in is more relevant to the question, and I haven't got enough room to write them both. I'll do breathing in, and then I'm going to do breathing in, and then breathing in, because it's more relevant to the question, which is bringing oxygen into the lungs. But breathe in. When you breathe in, what happens to your ribs? Put your hand on your chest and breathe in. Should be in the exam if you got this question. We want to say ribs move up, diaphragm moves down. And why is that? So, in fact, I probably should say intercostal muscles contract, diaphragm contracts. So, ribs move up. Oh my gosh. Diaphragm flattens. I'm just going to say moves down, right? Because you can say flattens if you want, moves down. What this means is we create a bigger space. By moving up and down, we create a bigger space. So we're going to say volume in thorax increases, pressure decreases. Volume and pressure are inversely related in this process. So as the volume increases, the pressure, the gas pressure inside the lungs decreases. It decreases below, and I'm going to write ATMS for atmospheric pressure, and then air rushes in down a pressure gradient. Now, you can talk your way through this. You put your hands on your chest. If you're in the exam, you're like, oh shit, I've forgotten what's happened when you breathe in. Put your hands on your chest, breathe in. If your ribs are going up, then the diaphragm is going down, because what you're trying to do is make a big space, because then the gas particles are really spaced out and you're lowering the pressure. And what this does is this causes your pressure in your lungs goes lower than outside. So air rushes in like the wind. It's the same way that wind moves. If wind's blowing through your hair, it's coming from high pressure to low pressure. So the air rushes into your lungs. Now, once you know that and you can track it all from that, then you can do the opposite. Breathing out, the intercostal muscles relax. The diaphragm is a muscle that relaxes. The ribs move down and in. The diaphragm moves up. It domes like this. It moves up. This decreases the volume, increases the pressure, so the pressure is above the outside pressure, forcing the air out down a pressure gradient. So how do you keep in your mind that contraction is breathing in and breathing out is relaxed? Think about meditating. When people say calm down, breathe out and relax. So breathing out is a relaxation process. And relax, because your muscles relax when you breathe out. Anywho, peoples, I don't think this is on the spec. I don't think this is on the specification. All right. So let's go. What do I want to do? I want to do the heart. We're doing organizatione. Let me go over the blood. So make sure you guys know the blood. Oh my days. This thing is effing with me. Stop doing it. Oh my good gosh. Poor Ishal, you've been spending... Ishal, you know what? Concentrate on your exams. You don't have to sit here. You can go revise, do whatever. You can turn people off so that you can't see their comments. You don't have to kick them out. You can just kick them out, but whatever. I should do that. Because otherwise you might be getting distracted from revising. So, let's go through. You need to know what each of these are. There are 4 components in the blood. So what is A? What is B? What is C? What is D? Yeah, they're all 4 are here, but I'm going to go through A. So A is red blood cells. Now, sometimes called erythrocytes. You do need to know, you might say, but sir, it's white. That's not the point. It's not why white blood cells are called white blood cells. Red blood cells have no nucleus, and from the side they look like a dumbbell, like that, like a jam donut. We call this shape biconcave shape. Large, it gives them a large surface area for absorbing O2. They have no nucleus. This is part of specialized cells as well. So they have no nucleus, so they can hold lots of hemoglobin. So more room for, I'm just going to put HB, but that's hemoglobin. So more room for hemoglobin to pick up oxygen. Okay, so that's your red blood cells. If we look at B, B is white blood cells. Now, white blood cells, I'll come on to with immunity. I'm not going to go over them here. They fight disease by phagocytosis, or lymphocytes produce antibodies that kill bacteria. C is plasma. Now, you've got to be able to name about 6 things that you find in the plasma. So vitamins, minerals, hormones, glucose, amino acids, carbon dioxide. There's some water, right? So there's at least 6, 7 things there. So it should be easy enough. D is platelets. They are broken bits of cell. Platelets clot blood and help cuts heal and scabs form. Who do we need to kick? Do we need to kick anyone? Okay. Right. Okay. Blood components, nice and easy. Let's move on. Now we're going to look a little bit at well done, well done, well done. Okay, now we're going to look at these. So what is F? What is G? What is H? Can you work out what F, G, and H are? What is G? What's F? What's H? Yeah, exam Q&A is very, very popular. You go on there. That'll be good for you guys. G is artery. F is veins. Well done. H is capillary. I have a nice evening every evening, dude. I think exam Q&A sell you courses as well, last minute courses if you want to part with some money so that you can, you can part with some money the night before the exam. I'm sure that will help. Not. Yeah, of course I'm better. I've been teaching for double the length of time of all three of them dudes. But that's okay. It doesn't matter. I'm just, what are we doing? What are we doing? Wait. So, check this. One cell. It's one cell thick. So H is a capillary because it's one cell thick. Remember, veins have, and you get 6 marker on this, veins have a large lumen, low blood pressure, and valves. They also have muscle and elastic, but arteries have a small lumen, so that makes high blood pressure, and they have no valves. And capillaries, as we know, they are permeable. That means they have holes in the walls to allow gas exchange, and this is where they're permeable. This is where oxygen, can go in or out, and carbon dioxide in or out through the gaps between the two cells. So they're permeable, they have gaps, and walls one cell thick. Okay, let's move on. Let's get off of that. Well, it's not quite the same. They have gaps between the cells. They're called fenestrations. It's not really that it's partially permeable. They do have a membrane and they don't have a cell wall, but so they do have a partially permeable membrane, the cells, but it's actually the gaps between they have gaps between the cells to let substances in and out as well. Okay, I'm zinging on. Now we're going to look at the heart. My good gosh. Oh my days. Anybody that I made admin, don't feel you got to hang around or anything like that, because you can chip off whenever you like. Right. Label the 4 blood vessels. So do you know what A, B, C, and D are, peeps? What is A, B, C, and D? B is especially probably the most asked for blood vessel that's connected to the heart. Yeah, so small lumen creates a high blood pressure so that the blood can travel to further around the body, and the larger lumen makes a low blood pressure. No, the pacemaker doesn't regulate the blood pressure. The pacemaker regulates the rhythm of the heart. Okay, so you've got blood vessels that lead to these. You've got the vena cava that leads here. And essentially, let me go through what happens here with the heart. So essentially, the blood comes into the right atrium, the blood goes down into the right ventricle, and then up through here and off to the lungs. Now the lungs, dodgy looking lungs, at the lungs, the blood is going to turn red because it's going to pick up, well, it doesn't really turn red, but you get the thing, right? The idea. So it's going to get oxygenated. It actually comes back in. A big mistake, people think it comes in there. It comes in this side through the left atrium and then into the left ventricle, and then it goes up through the aorta, which is C. It goes round the back of the heart and it shoots off to the body. Okay, and this is known, this one over here is Bosh, Bish, Bash. This, I'm just going to put PA. Oh my days, look at this sickness. That is pulmonary artery. This is the pulmonary vein. And this over here is the aorta. Now, these are very typical questions. In which chamber is the pacemaker found and what is its function? So the answer is the right atrium. And it regulates the rhythm of the heartbeat. So, regulates rhythm of, oh my good gosh, of heart. I should put heartbeat, but I'm running out of rules. Why is the left ventricle thicker? Who said, what is, yes. So the left ventricle is thicker, more muscle, but it's a 4 marker. Why is it thicker? More muscle. You need to say greater contraction. I ain't got the room. Greater contraction. More force or pressure. So blood goes further. You must mention distance. So blood goes further. I should just talk because I can't put that on there. So blood goes further. What are the valves for? So that's a 4 marker. Greater muscle contraction, more force generated, blood can travel further. Now, they could ask you, why is the right thicker than the atrium? Why are both the ventricles thicker than the atriums? Because they're both sending the blood further. This is sending the blood to the lungs. This is sending the blood to the body. They need to have stronger force of contraction so that they can create a higher pressure and send the blood further. The atriums only need thin walls because they're only sending the blood into the ventricles, just a couple of centimeters. Now, what are the valves for? They prevent backflow. So you do need to know these prevent backflow. And then for the last bit, I think Keishan put up the perfect answer there. What is the answer if there's anything wrong with the heart? If they say the valve's broken, there's a hole in the heart, so-and-so's heart doesn't work properly. What's the issue? The whole issue is we won't get enough oxygenated blood to the cells of the body. So less blood flow, less oxygen to the cells, less aerobic respiration, less energy released, cells die. You can say anaerobic respiration happens, lactic acid produce pain and stuff like that. Cells fatigue, cells tire, muscles tire. So you can do that. It doesn't matter what the error is with the heart, it will always result in less oxygen to body cells, less aerobic respiration, less energy released, and then more anaerobic respiration, more lactic acid produced. Cool. Pulmonary artery is the only artery in the body that carries the oxygenated blood. Arteries, think of the A, is always away from the heart. That's the same. There's no artery that goes, yeah. What are we doing? We already did this. Okay, cool. Let me go through. Let's do some bits. What else? Okay. Which one? Cam in Jenny, what do you want me to write? What are the benefits of a double circulatory system? Okay, so cat aviation, double circulatory system, that is something that you need to know. The double circulatory system is 1 mark normally, sometimes 2 marks. So I'm going to give you the 1 mark answer for the double circulatory system and then the 2 mark. The double circulatory system, if you're asked what it is that the Lord, someone said Lord, so left side of the heart pumps oxygenated blood to the body. right side of the heart pumps deoxygenated blood to the lungs. If you can't remember that, you will get 1 mark for saying right side pumps blood to the lungs, left side pumps blood to the body. And that is why we call it a double circulatory system. Now, being a large organism, the double circulatory system is more efficient because we get to repressurize, we get to add extra pressure when we want to pump it to the body. If we had a single circulatory system, then you have to go through the capillaries in the gas exchange place, which is the lungs, in fish it's the gills, and then you have to somehow make it around the whole body. So it's less efficient. So we can generate higher pressure by sending the blood to the lungs, then back to the heart and pressurizing it again to the body. So we get greater pressure. If there is any fault in the heart, Genevieve, the answer to faults in the heart, let me lick this one down. Now, I'm not saying that there aren't other things, but I'm just trying to give you the generic answer that will always be part of the mark scheme. This is a generic 4 marker that will result from any fault in the heart. Think about it. If I said, oh, there's something wrong with your car, with your dad's car or your mum's car, what's the result? Well, the result is you don't get dropped off to school, right? If there's something wrong with a car, that's the result. Anything wrong with your heart, what's the job of the heart? It's to pump blood with oxygen to cells so they can do aerobic respiration. So, faults with heart, and even if you don't, what I'm saying is even if you don't understand what is going on about, oh my God, that is just disgusting. Even if you don't understand what is going on about, so I'm just going to say like heart problems, right? If you don't understand, this is always on the mark scheme, these things. And then I'll explain. There are nuances and other bits for the different faults, but I'll explain. Right, heart problems, you're going to get less blood flow to cells, or even better, less oxygenated blood. Better to say that, less oxygenated blood to cells, which means less aerobic respiration. You must say aerobic. It's not going to affect anaerobic because anaerobic doesn't need the blood. It doesn't need oxygen. Maybe it does need glucose though. Less aerobic respiration. Oh my good gosh. Why do we do respiration? Energy. So less energy. Don't say energy made. It's actually released because respiration releases energy from glucose. It doesn't make the energy. Less energy released. That should be a D. Less energy released. And then what does that entail? We can just say cells die. Now, if they're muscle cells, they won't have energy to contract. And then you could go on to say more anaerobic respiration. You could literally go on for hours with this. More anaerobic respiration. And that's going to mean lactic acid. What do you know about lactic acid? Lactic acid is toxic, damages cells, causes tiredness, fatigue, and pain. So you could add that all in as symptoms of when the stuff's not going. Right, now, specific heart problems. If the valves don't work, valves prevent backflow. So I would just add to this. If it said, oh, you've got a problem with the valve, 6 marks, how does it affect the heart? I'd be like, well, a valve prevents backflow. So actually, we're going to get backflow. Blood is going to go the wrong direction. This is going to mean that we get less oxygenated blood to cells, less aerobic respiration in the cells, less energy release, cells die. Now, if it said there's a hole in the heart, between the septum, the middle of the heart, between the ventricles or atria, then I'm going to get oxygenated and deoxygenated blood mixing. If the blood mixes oxygenated and deoxygenated, when the left side is supposed to pump oxygenated blood, it's actually pumping some of the blue blood, the deoxygenated. And guess what? That's going to mean less oxygenated blood to cells, less aerobic respiration, less energy release, cells die, more anaerobic respiration, more lactic acid. So if I say you have coronary heart disease, and you get fatty deposits which block the coronary artery supplying blood to the heart, guess what? That's another heart problem. But this time it's directly about the muscle in the heart. But I'm still going to say less blood flow, less oxygenated blood to cells, less aerobic respiration, less energy released, cells will die, more anaerobic respiration, more lactic acid, tiredness, pain in the heart, all of that. So It can be anywhere in your body. It could be to your foot. It could be to your heart. With coronary heart disease, it's specifically talking about to the heart. So it doesn't really matter the problem. That's always going to be part of the mark scheme, but there might be other things on there as well. Specifically, like if it's valves that are broken, they can't do backflow. If it's a hole between the heart, in between the chambers, you're going to get a mixing of deoxygenated and oxygenated. Now, if there is backflow and you get less blood to the lungs, deoxygenated blood to the lungs, that means you're going to pick up less oxygen. So you get less oxygenated blood, then you spit that bit there again. But it doesn't really matter. That's always going to be on the answer. But you might have to adjust it depending on which bit of the heart's wrong. Right, we need to talk about coronary heart disease. Okay, look, they've never really asked about saline solution, but artificial blood is like salt and water. You put you on a drip, and the idea is to keep your blood pressure high so your blood is pumping. In the future, we hope to have a chemical that mimics hemoglobin and can pick up oxygen for us and deliver it. So if someone has lost lots of blood through an accident, but if they've lost too much blood, you have to have a blood transfusion. So instead of giving you salt water into your veins on arteries, they don't actually ever put it into an artery because that would kill you. They put it into a vein because it's low pressure. Yeah. Now, coronary heart disease we need to talk about. There's many risk factors for coronary heart disease. So you can talk about poor diet. What do we mean by poor diet? We mean high saturated fats or high in cholesterol, high in salt. All of these will lead to artery damage, which will mean fatty deposit, and then that answer that I just said about the less oxygen, less aerobic respiration, less energy. So obesity is linked with it. Your genetic, people just have heart disease in their family. Your gender, men are more likely to have heart disease than women. Stress, so people who do stressful jobs have been shown to suffer from heart disease more. Smoking, all of these things are called risk factors. Now, some of them are lifestyle, and that means you can choose. A lifestyle factor you can choose. Apple, donut, you choose. Your genetics, you can't choose. You're born with those. Your gender, yeah, you can't really choose those factors that are going to affect your heartbeat. There's other lifestyle ones you can choose, like your diet, whether you take drugs, whether you smoke, whether you do a stressful job. Those are lifestyle factors. Now, most risk factors increase your blood pressure, like high salt and stuff, and high blood pressure is a massive risk factor in CHD, okay, so in coronary heart disease. And coronary heart disease means less blood flow to the heart, and then you get that list that I just went through. Okay, what else? We want to go to heart transplants. So we need to talk just a little bit about heart transplants. Heart transplants are essentially, you can have a live biological one or a metal or plastic one. Anything metal or plastic that is put in your body has a danger of getting blood clots. So just have that as a general rule. Anything metal or artificial that's not made of living tissue, the risk is blood clots. Anything that's living, the risk is rejection. Your white blood cells do not attack metal and plastic because metal and plastic do not have proteins on, called antigens, that your white blood cells can recognize. So if you give me a metal heart, white blood cells will not attack it. If you put a stent in me, a metal stent, white blood cells will not attack it. They don't, it's not a living thing, so they're not on the lookout for that. However, the danger is blood clots and risk of infection at surgery. Biological things, the risk is rejection. So we will get onto that. So we're going for coronary heart disease a little bit. Okay. White blood cells sometimes recognize donor heart tissue as foreign and they attack it. This is called rejection. Your immune system creates antibodies that then lock on, attack, and destroy tissue like heart tissue, liver tissue, lung tissue, whatever you've had transplanted, okay? It's much better to have therapeutic cloning, have the organ made by therapeutic cloning, then you won't have a chance of rejection. In order to reduce the chance of rejection, people who have donor hearts are often given immunosuppressant drugs. So this hasn't been tested on the exams yet but I just went through that. Yeah. So this was just the artificial hearts, metal and plastic, less chance of rejection because they're not made of living tissue. However, blood clots can form and that can lead to a heart attack. If you have artificial blood, you've got to take blood thinning medicines. If you have an artificial heart or something, you have to take blood thinners. If you have a stent, you have to take blood thinners. Anything plastic or metal in your body, you will have to take blood thinners to prevent blood clots. Blood thinners are a type of medicine. Discuss the advantages and disadvantages of an artificial heart being fitted. 6 marks. Yes, you could say therapeutic cloning to reduce rejection, but I don't know if it will come up in that way where it would be no problems. Yes, it's therapeutic cloning. It's an embryo stem cell, but it has your own DNA or genes in it. Now for this one, have a look. I'm going to put the answers up, guys. So this is the answers for artificial heart is less likely to be rejected. You do not have to wait for a donor because they can just make you the heart. You can keep the person alive. Obviously, that's a good thing, right? Both of them keep you alive. Against, it's made of plastic and metal, so it can result in a blood clot. Surgery means you could get an infection. You could suffer bleeding during surgery. Electric motor, it might break. You have to have blood thinning drugs for the rest of your life. Okay, and blood thinning drugs mean that you bruise easily, and if you get a cut, you could just bleed to death. The reason is blood thinners get rid of your platelets so that your platelets don't clot your blood. And the reason is to avoid you getting a blood clot. If we did the advantages or disadvantages of a biological heart, it would be slightly different. You still need an operation. You could still get an infection, but rejection would be the thing we want to talk about as a disadvantage. Obviously, the advantages are that you won't get the blood clot. Yeah. And that you were kept alive, right? Okay, we've got to look. This comes up quite a bit. Yeah, biological can be rejected. Coronary artery. Coronary artery is the one that feeds the heart oxygen to keep the heart muscle. As soon as the aorta leaves the heart, taking oxygen on the journey around the body, a little branch of it called the coronary artery comes off and feeds the heart, delivering oxygen. So you need to know stents versus statins. So make sure you can do a 6 marker on stents versus statins. Stent is going to have all the problems of artificial stuff. Blood clots, you need to have blood thinners. What's the good thing about it though? It keeps the artery open, it increases blood flow, more aerobic respiration, more energy released. You get the idea, right? Better contraction of heart muscle. What's the difference with statins? Statins are a drug. You have to remember to take them. Stents you don't have to remember to take. Statins, you don't need an operation, so you won't get an infection at the site of the operation. But statins have side effects, like all drugs. They could have side effects which could damage your organs, like your liver and stuff. Biological valves do eventually wear away and need to be replaced. I see one said that, but mechanical valves do as well. They both will eventually get problems. Biological valves, I'm not sure which one lasts longer. Maybe biological valves last longer, actually. Biology tends to be better than machine at the moment in terms of length. Statins have side effects. They can include liver damage. They can even be fatal. You can die from statins, like in very rare instances. Now, stents require you being chopped open. Whenever you're chopped open, viruses or bacteria could get in. And you could also get a blood clot called a thrombosis at the site of the stent. But that can happen with any operation, so with a heart transplant or anything. Remember, what is coronary heart disease? It's when fat, cholesterol, well, it's not really cholesterol, LDL cholesterols increase the chance of this happening. These low density lipoproteins, these LDL cholesterols, they help fats build up inside your coronary artery and they reduce blood flow. So they reduce oxygen to the left ventricle of the heart. The left ventricle pumps your blood around your body. If it has less oxygen, it cannot respire as well, aerobic respiration, release energy and contract as hard. And then these are just statins called, yeah, cool. That's a stent. You know what that's called. It's like a cage. They insert it with a water balloon. All right, have a go at this one, people. See if you can write down, or just do it in your head. Quickly talk through it. Statins and stents are 2 treatments used to reduce the risk of someone having a heart attack. Evaluate the use of statins. Compare with the use of stents to reduce a heart attack. This is a past paper 6 marker. We're nearly at the end of this topic, and then we're going to have another little break. Oh my God, I'm going on till 10 o'clock. OMG. This is discussed in live. All right, peeps, I'm going to put the answers up. So just make sure you could do this. This is the 6 marker. So challenge yourself. Of course, you can pause if you don't want to see the answers. This would be the answers. So statins and stents are 2 treatments. They use blah, blah, blah. Indicative content. So really you want 2 and 4 or 3 and 3. You don't have to have exactly 3 of that and 3 of that. You could have 2 and 4, etc. Easy to take. No operation, that's what invasive procedure means. No operation needed. Decrease the blood cholesterol, slow down the buildup of fatty materials, maintain blood flow, low cost compared to stents. Disadvantages of statins are side effects. Yeah, you might forget to take the drug. Now stents, maybe they don't have stents on there. Oh, compared with the use of stents. So with stents, I don't think I got the stent bit to it, but yeah, the indicative content for stents, if you were talking about stents, would be that they reduce the risk of heart attack because they increase blood flow by opening up the coronary artery. You don't need to remember to take them like you do with statins, and they don't have side effects like statins because they're not a drug. But the downsides with stents are that you could get a blood clot, so you'd have to take blood thinners, which is a risk of excessive bleeding or bruising. And you could get an infection at the site of the surgery. Okay. Oh my days. What's happened here? Okay. Oh my gosh, people. You got to know that. You got to be able to label that. Is everyone all right with leaf structure? Is leaf structure okay? Is leaf structure okay, folks, for you? Or do you want me to go through leaf structure? I've got to go through leaf structure and cancers, and then I'm going to take a quick break. Oh my gosh, this is so long. I don't know why I've waffled so much this year. Normally my previous ones haven't been this long. I don't know why I've gone on a waffle. I've done a waffle job this year. Okay, so just remember for this, you got waxy cuticle. This is not actually cells. This is a chemical produced by the upper epidermis. The upper epidermis is transparent. Okay, let's lighten. So this would come under explain how the structure of the leaf allows photosynthesis to happen. So it's transparent. Let's light in. So your light is going to come in here through the upper epidermis into these cells here. This is a tissue because it's made of the same cells. And you can call them palisade cells or palisade tissue. So this is palisade mesophyll if you want to get excited. Palisade tissue. These cells, palisade cells are tightly packed. That means there's no gaps. They don't want to let the light through. They're tightly packed. And lots of chloroplasts. Lots of chloroplasts for absorbing sunlight. For absorbing sunlight. Oh my gosh. I've got questions on all of this. Spongy mesophyll. Do you know what spongy mesophyll do? Hopefully you do. Make sure you test yourself. Spongy mesophyll. These guys have got air spaces, which allows gas exchange. You can say diffusion, if you like, of carbon dioxide or oxygen. And then we've got a couple more. We've got the lower epidermis. And the lower epidermis contains what? The lower epidermis contains guard cells, which open and close stomata. So they can open and close your stomata, right? These little holes here. The stomata are the holes. And then I've missed out the vascular bundle, which is the xylem and phloem over there. I haven't got room for writing xylem and phloem. It's called the vascular bundle. The xylem transports water and mineral ions, and the phloem transports glucose. Easy. Okay, what we're going to do, I'm going to zing onto this now quickly because okay. So, you need to be able to compare the xylem and the phloem. Xylem transports water and minerals. It's part of transpiration stream. Oh, do you know what? Let me just do a verses. Let's do a verses thing, right? Let's do a verses. Oh my good gosh. Let's rub that all out. Oh dear, oh dear, oh dear. Okay, what I'm going to do here, right, I'm just going to keep it simple. This is made of dead cells. Where are we? Come over here, mate. Get over there. This is made of living cells. This is involved in transpiration and one direction, Harry Styles only. And this one is translocation, which is the movement of glucose and amino acids, two directions. It goes up and down. It transports amino acids and sucrose, sugars and food, dissolved sugars, yeah. So let's just go transports sugars. It also transports amino acids in the exact same way. This transports, I'm just going to say water, but yeah, there are minerals dissolved in water. So we've got dead cells, living cells, transpiration, translocation, one direction, two directions, transport water, transport sugars. This has lignin in cell wall, lignin in cell walls. Okay, lignin for extra strength. I think that's about it. Awesome. Now, Bosch. Something else you guys have to know is the difference, which comes up occasionally, so just watch out for this one, all right? The difference between transpiration and the transpiration stream. So I'm just going to quickly smash that through for you. No, don't do that, you idiot. Come on, Tom, you prat. What is the difference between transpiration and the transpiration stream? So, transpiration is evaporation of water from stomata in leaves. All right, so transpiration is just how water escapes from the leaf, whereas transpiration stream is water from roots up xylem and then out leaf. via evaporation or transpiration. Okay, so the transpiration stream is something different. It's talking about how the water comes into the roots by osmosis, goes up the xylem, and then out the leaf. So it's like three things like that, and we call it a stream, like a river, whereas transpiration is just a word to describe water as evaporating from the leaf. Just watch out in case you get nuts. Right, oh my good gosh. Yeah, I better get on and do non-communicable and non-communicable diseases. Well, I'll just say, before I do non-communicable and communicable diseases, you do need to know about cancers. So the last thing I'm just going to say about cancers, and then I'm going to get off this topic and I'm going to do communicable and non-communicable diseases. Cancers can be malignant or they can be benign. Benign means good. There's no tumours that are good. There's no cancers that are good. So I should say tumours can be benign or tumours can be malignant. What do you need to know about each? Malignant tumours can spread around your body and invade other parts of your body. So I could have a cancer in my lung and it could spread to my liver and invade my liver and damage my liver too. If it's a benign tumour, it does not spread around your body and it does not invade organs. It's kept in a sac called a capsule. And benign tumours do grow, but not as rapidly as malignant. So both tumours have uncontrolled growth via mitosis, so they're just copying themselves and growing and growing. So you do need to know that tumours have uncontrolled growth, malignant tumours have very rapid uncontrolled growth, and they can invade organs and spread around your body. Translocation, no, translocation is no. Lyle Mackey's translocation is the movement of sugar and amino acids, not minerals. Minerals are moved up the xylem to the leaf via transpiration. So amino acids are broken down proteins. Minerals are tiny things you find in the periodic table in chemistry, like phosphorus, magnesium, lithium, sodium. You get the idea. They're elements from the periodic table. Generally, you have nitrates as well. They travel up the xylem in the transpiration stream. So it's not just water. It's the movement of water and minerals. You could include the minerals. Cancer is paper 1. Dissolved sugars and amino acids. Cool. What else? Was there anything else in organisation? Let me get off of organisation. We're going to go and do some... we'll go and do some communicable disease. What the ass? We'll do some communicable diseases. So, I'm going to take a break in a minute, but... Okay, the Grid Box Podcast. Transpiration is just the evaporation of water from the leaf. So here's my leaf. Water... Oh, let me go... I was going to draw it, but yeah, we don't want to see one of my crusty drawings. All right, let's do a crusty drawing quickly for you. It'll take 30 seconds. Oh my good gosh. Okay, so we got the crusty leaf. This is the difference between transpiration and the transpiration stream. Transpiration is the evaporation of water molecules from the leaf. That is known as transpiratione. Let's write that down quickly. So this is transpiration. Can't really see that. That's what you have when you've got a stupid background like me. And then the transpiration stream, we're going to draw the rest of the plant and we're going to do its little roots. The transpiration stream, if you're asked the difference, is how water moves into the roots. So H2O moving into the roots by osmosis into root hair cells. Then it travels up the xylem. And then it is also evaporated out of the leaf. So the transpiration stream is the whole thing. So if you're asked what the transpiration stream is, you need to say it's water going into the roots, up the xylem, into the leaf, and evaporating by transpiration through the stomata. If you're asked about what is transpiration, it's just water leaving the stomata. That's the difference. So it's just a technicality, a technical thing. Right. Communicable disease is a disease that can spread. Cancer can't spread. Diabetes can't spread. Obesity doesn't spread. So a disease that can spread from person to person. Bush, bish, bash. What do we want to go through very quickly? What piece of equipment do you need? Yeah, microscopes. Communicable disease. What are they caused by? Pathogens. Do you know your 4 types of pathogens? That's right, the Grid Box podcast. But if you're describing the transpiration stream, you must say evaporation of water from the leaf. So you're going to say the whole thing. Transpiration stream's the whole thing. It's the movement of water through the plant from start to finish. Protists, bacteria, fungi, virus, boom! Well done. Right, now we need to know, how does each one of those make us ill? So how do protists make us ill? How does fungi make us ill? How does bacteria make us ill? How does viruses make us ill? They might say to you, how does salmonella make you ill? And the answer, peeps, is right here. So bacteria always make us ill, whether it's salmonella or gonorrhea, whatever is a bacterial disease. They make us ill by producing toxins. Toxin is the word that links to bacteria. Fungi grow things that go through our skin. They're called hyphae. They penetrate our skin. Protists are parasites. That was crap labeling. And viruses, two things you want to say about viruses. They reproduce rapidly. They're inside our cells, not outside. They're inside our cells and they cause them to burst. Boom. Right. You do need to know how pathogens spread. Pathogens can spread from dirty water through droplet infection going, achoo! They can spread through contact, door handles, indirect contact, sharing towels, sharing sandals. All of those things are different ways pathogens can spread. Dirty water, droplets, droplet infection, direct contact, indirect contact, sharing towels, sharing sandals, sharing a needle if you're a drug addict, etc. Right. Let's go through some of the different ones that for the AQA Biology spec you have to know off by heart. Well, hopefully you get more than a 6-7. Right, what I want to say is symptoms of measles. Oh, sod this. Measles is caused by a virus. You get a red rash. You get a fever. Why do we vaccinate everyone against measles? So you need to know measles is caused by a virus. You could be asked that. Often that's the most common thing about measles, that it's caused by a virus, and that's a one marker. But you might be asked for the symptoms. Red rash and fever. Easy to guess, right? Measles is red spots and you get a fever. It can be fatal, which is why we protect all babies by vaccinating them at birth against measles. We give them the MMR jab, which is measles, mumps, rubella. Right. Now, you could get, what type of pathogen causes HIV? What does the V stand for? What does the V stand for? Virus. The V is for virus. What does it do to your immune system? It destroys your immune system, specifically white blood cells. So it's going to blow up your white blood cells. Viruses make cells explode, but the bad thing is that HIV lives inside your white blood cells, which are your defenders. So it kills your defense. And then you die from a cold sore, or something else. Right. Is there a vaccine for AIDS? Can you get vaccinated against AIDS? No vaccine for AIDS. The reason is because it changes its proteins. It basically evolves and mutates. It has a high mutagenic rate. Right, we have another virus you've got to know about. The third virus is the tobacco mosaic virus. They will call this TMV as well. TMV. Right, this infects many plants. Look, you can see the picture there. Okay, like I said for heart disease, for plants, there is the same answer all the time. What is it that plants do? What's the number one thing you know about plants? The word is photosynthesis. So any disease is going to screw up photosynthesis. So we're going to say less photosynthesis. What does photosynthesis make? It makes glucose. So if we have less photosynthesis, less glucose, less respiration, less ability to release energy, less ability to grow, make protein, do all of that stuff. So you just follow the process. It doesn't matter what the plant disease is. It's always going to mash up photosynthesis. Now, how does it do that? If you want to get a bit more excited, well, what's the green stuff? The green stuff is chlorophyll. So it destroys, all plant disease is going to destroy chlorophyll. Means you absorb less sunlight, you do less photosynthesis, you make less glucose, you can do less respiration, therefore you can't grow. And that's all you got to do with any plant disease. Right, let's zing on rose black spot. So we got two here. These are fungal diseases. Now, with TMV, with viruses, fungal diseases are bad because they can spread through the air because fungi reproduce by spores. So rose black spot obviously causes black spots on leaves. Athlete's foot makes your skin itch on your foot and fall off. Treated by removing and burning infected leaves. Well, you're not going to burn your feet. Spreads through the wind. Spread through sharing towels and sharing your sandals. Treated by special fungicides between the toes. Yeah, that one. There we go. Right. Normally with fungal diseases with plants like rose black spot, you're going to burn them. Listen, rose black spots do the same thing as TMV. It just has black spots instead of yellow spots. It's going to destroy the chlorophyll, which if you have no chlorophyll, you can't do photosynthesis. You can't absorb light. You can't make glucose. You can't respire. You can't release energy. You can't grow. Bosh. Suggest? Can you suggest how... Yeah, I've just done that. Right, that's the answer for that. Good. Right, I want to speed up. So malaria. Look at the two pictures of the mosquito and answers the question. You don't need to know if it's an invertebrate or vertebrate. You won't get asked that. They're invertebrates. They don't have a spine. Anyway, yeah, you don't need to know how it's an insect. Oh, what's all that? That's a rubbish picture. Okay. Mosquitoes are vectors. VECTOR. They carry the protist inside them, but it doesn't harm them. How can you stop mosquitoes? 3 ways. Mosquito nets. So this lady here is sleeping under a net to stop her getting bitten by a mosquito. By using insecticides. CIDE means death. Insecticides will kill them. And by sterilizing the mosquitoes, you capture the men and you destroy their sperm. You release them into the wild. The male mosquitoes go out. They have sex with the female, sexual intercourse. The females go, woohoo, we're pregnant. And they fly off to do wherever pregnant mosquito females go. They go to, like, you know, chill out and have their babies, not realizing that the men are firing blanks. They've been made sterile. Their sperm are dead. And then what happens is when the breeding season's over, there are less babies born because half of the women think they're pregnant, they're not pregnant, and da da da. Now, do you have to explain all of that? No, you just say sterilize male mosquitoes or sterilization programs. So, or disrupt the mosquitoes' breeding. So those are the ways. It's better than capturing the men and killing them, because as long as you, even if you leave just a couple of thousand male mosquitoes alive, you don't kill all of them, then they're going to go around and impregnate all of the women. So you must sterilize the men and release them so that they don't reproduce properly. Anywho, right, that's mosquitoes. Now, that's a protist, by the way. Salmonella is more likely to spread in caged chickens because they're close together, free range further apart, so it can't spread so much, less contact. Poultry is turkey, chicken, pigeon. You can say stop the mosquitoes breeding. That's right, Lucas. Why is it extremely dangerous to not cook chicken properly? So it's dangerous not to cook chicken properly because chickens have salmonella. About 50% of the chickens in the UK have salmonella on their skin, and cook them properly and you will kill the salmonella. Wash your hands. Salmonella, they'll often say, if you've got salmonella, how do you stop it spreading? With any disease, isolate. So you stay in your bedroom, you get your food pushed under the door, whatever. Yeah. All right, no. But isolation, wear a mask, wash your hands. Salmonella won't spread by droplets, so wearing a mask won't help with salmonella, but it will with like measles or something, because measles spreads by droplets. What else? What else? You can vaccinate the chickens. Salmonella is caused by a bacteria, and bacteria are treated with a drug. What drug kills bacteria, guys? It is bacteria, salmonella. What drug kills bacteria? Do you know the drug that kills bacteria? And do you know who invented it? No. They're going to look at all your answers. If you put incorrect answers, then they may take marks off if they contradict each other. Like if you're talking about salmonella and you're like, yeah, salmonella is caused by a bacterium, and then later in the answer you're like, yeah, because it will stop the virus spread. Now you've referred to it as a virus. They're taking your mark away. You don't get the mark for calling it bacteria. So that's a contradictory answer. But if you said something like that is not contradictory but is not necessarily correct, they'll ignore it. Now, salmonella, you can vaccinate the chickens. You can't vaccinate humans. Far too expensive. Waste of time. You just got to cook the chicken properly and wash your hands, and then you won't get salmonella. Okay, so eat cooked chicken, eat cooked turkey, eat cooked birds. Poultry means birds, and wash your hands, and you won't get salmonella. You do need to know the symptoms. Do you know the symptoms? Yes, antibiotics. First one was penicillin, and discovered by Alexander Fleming, and they will kill bacteria diseases like salmonella. Lucas, using a gas syringe is better, but you can count the bubbles over time. Unless it says what's a more accurate, better way would be a gas syringe. It's much more accurate. Okay, fever, vomiting, diarrhea. Excellent, Blaze. You got it absolutely spot on. Yeah, disinfect surfaces. Yeah. So, yeah, disinfect the surfaces, and yes. Right. Gonorrhea. Guys, gonorrhea is also caused by bacteria. So any bacterial disease, salmonella or gonorrhea, you can treat with antibiotics. That's why I mentioned them. If you've got a bad case of salmonella or gonorrhea, what medicine can you use to cure it? Antibiotics. Woo, woo, woo. The symptoms of gonorrhea are pain when urinating and green and yellow pus from your penis or vagina. And of course, use condoms or contraception. The pill, estrogen pill, will not protect you, but condoms or barrier methods will protect you. Okay, some questiones. See if you can do these two. What type of pathogen causes malaria? Protis. Boom. Nice. Man has the following symptoms. Yellow discharge from his penis, pain when urinating. The man has a bacterial infection. What is the most likely cause of his symptoms? Gonorrhea. Bruh. Easy. Right, let's go. Okay, eating food containing salmonella bacteria can cause illness. Two symptoms of infection of salmonella are vomiting and diarrhea. What causes these symptoms? They just gave you a clue here. Do you remember early on we went over what is it that all bacteria release? What do all bacteria release? So the salmonella ones cause vomiting and diarrhea, but the gonorrhea ones cause green pus from your penis or vagina. It's toxins. So bacteria always release toxins. Now they're not all the same. They cause different symptoms. Give 2 ways a person with a mild infection of salmonella can help prevent the spread of bacteria. So I don't think they're saying take antibiotics. So antibiotics will not be the correct answer here because they're saying it's a mild infection. How can they not spread it to other people? Wash hands. Awesome. No. So you can take antibiotics for salmonella, but because it's a mild infection, they would say only with a serious infection. Because remember, we're trying to not overprescribe antibiotics. If we do that, we encourage the formation of antibiotic resistant strains of bacteria. So wash hands, we could say isolate. Yeah, something like that. Right, let's zing on. A person with AIDS may take longer than a healthy person to recover from salmonella. Why? Why does a person with AIDS take longer to recover from another infection like pneumonia, salmonella? So what do we reckon? Weakens the immune system. Good. Yes. So weakens the immune system. Good. Boom. Okay. Now that means, oh, come on, Tom. Sort it out, mate. I'm going to say less, oh my gosh, less WBCs, white blood cells, shorthand. Don't write that in your exam. Less antibodies. So I'm just giving a couple of reasons. Like you're going to have less antibodies. I'm going to say less phagocytosis, right? If you've got less white blood cells, you're going to have less antibodies and less phagocytosis, less B cells and less phagocytes. Salmonella bacteria can be transmitted from chickens to humans. Chickens can be vaccinated to prevent the transmission of salmonella bacteria to humans. Suggest one other way farmers can prevent the transmission of salmonella from chickens to humans. They already said to vaccinate. So other than vaccinating their chickens, boom, clean the food, wash the chickens. I reckon give them antibiotics. It's actually what lots of farmers do, which hasn't helped because it increases the formation of resistant strains. Give them antibiotics. I'm going with bish bash bosh. Rose black spot is a disease. We went over this. What causes rose black spot? Yes, spread the chickens out. Nice. Good. Yes, spread the chickens out. Fungus. Well done. Who said fungus? Blaze. Absolutely blazing. Right. Oh, geez. What happened there? Hit the rubber. Okay. My good gosh. Right. I'm going to do these for you. You can have a look. And I'll do these. HIV can increase the chance of an infection such as ammonia. So because it lowers your white blood cell count, it will increase your chance of catching any other disease. Malaria includes the life cycle of an insect. Basically, the key word there is insect. That's going to draw you to that one. Salmonella spread by not washing hands thoroughly. Yeah, can be spread like that. Gonorrhea, sexually transmitted. What are the symptoms of gonorrhea? 2 boxes. Food poisoning is definitely no joke. I had salmonella once when I was 18, many years ago, many moons ago. Yellow discharge, pain when urinating. That might say green discharge sometimes. Okay. How can we control malaria? I went over this earlier. What are 2 ways to control malaria? About 108. That's how old I feel right now. I'm 50. I'm 50. Yeah. Or thereabouts. There or thereabouts. Yeah, 108 maybe. 2 ways to control the spread of malaria. So we're going to say mosquito nets. We're going to say sterilization programs. This is the problem. I can't spell programs. All right, let's lead it. Interrupting breeding. I'm just going to say insecticides. Insecticides are like sprays, deodorants you can put on, little candles you can burn, mosquito nets, prevents them biting you. Nice. Okay, let's zing. What methods can be used? Okay, these are just methods. Washing hands. We've done these. This is like antiseptics. You can call these disinfectants. They basically kill microorganisms, but you can't take them. They'll kill you. That's isolation. Okay, so this is just some general terms. This person here is spraying with fungicides. He's killing like rose black spot or something like that. Masks. Masks won't stop all diseases. They won't stop salmonella. They won't stop gonorrhea because that's spread by sexual intercourse. It's an STI. Masks will stop things like measles or the flu, which are spread by droplets. Obviously, boiling water will stop the spread of cholera and ones from infected water. Okay, let's not worry about that. But one thing you do have to know here actually is, I don't want to go on about it too much, but your respiratory system. Right now, as you're watching this live, there are bacteria and viruses going into your mouth and down into your respiratory system. But luckily for you, you have cilia. They're not the same as villi. Don't get them mixed up. Cilia are little hairy cells with little hairs on, and they waft bacteria and they produce mucus, which trap the bacteria and viruses. Mucus is snot, basically, and that wafts up into your mouth. And every minute you swallow, even if you don't think about it, but you do it every minute of your life. You are swallowing bacteria and viruses and dunking them in stomach acid in your stomach. Anywho, I don't want to spend too long on that. Yeah, skin, cilia, stomach acid. 3 ways white blood cells defend against the body. Yeah, they're non-specific defense. You want me to go through the defenses? Okay, if I go through the defenses, then we have skin. Now that's a barrier. That's a barrier. You're not going to get asked about that or that. Oh my God, that does that look like the nose? You don't have mucus in your head. Jeez, that's terrible. So you got mucus in your respiratory system. I don't know why I've done that one there. That's stupid. But you got mucus inside your vagina if you're a woman. You've got mucus inside your anus. Are you going to get asked about that? Probably not. You got mucus in there as well, guys. Guess what? You got earwax in your ears. That's right. Any hole in your body is a possible entry for pathogens. So you're going to have something. Now, do men have mucus in their penis? No, because we have urine. When we wee out of our penis, the urine cleans and is an acid. It kills bacteria. So there is a liquid or a solution in every part of our body that traps and kills bacteria, like earwax. You've got tears in your eyes. They're salty. They're going to dehydrate and kill pathogens. So any entry point. Skin, obviously a break in the skin from a cut, so you'll need a scab to protect you. You've got stomach acid if something gets into your stomach. These are all non-specific. They'll kill any pathogen. They will just mash them up. Now, in your respiratory system, you need to be aware that we have cilia cells. Now, ciliated cells, they don't have a cell wall, but they have little hairs. What happens in your respiratory system is mucus is released. It's normally clear mucus. It's only green when the enzymes leak out of a dead pathogen, like a bacteria. But it will trap a little bacteria in it, and then that will get wafted into mouth. So this is cilia, these hairs, and this is called a ciliated cell. It's actually a ciliated epithelial cell because it's lining your respiratory system. This wafts mucus with trapped bacteria or pathogen wafted into the mouth. What do we do when it's in our mouth? We swallow. This happens every minute you're swallowing. We swallow and then the hydrochloric acid kills it. So I swallow just now after I finished speaking. Anything that got in my mouth while I was speaking has gone into my stomach, been mashed up by my hydrochloric acid, enzymes denatured. Oof. Okay. I'm gonna have to go get a drink, guys, and then I'm gonna do this bit. So let me give you a couple. I'm gonna come back to there. Let me just give you a couple of. I'm just gonna go grab a drink, and then I'm coming back. I'm gonna give you some questions to do while I get a drink. Okay. Have a go at these questions. I'll be back in 2 secondos. Okay, let's rinse through these and then get on, crack on, crack on. When athletes train hard, they can respire anaerobically. I don't think I actually need my glasses, do I? Lactic acid and carbon dioxide are produced. Oh gosh, what am I on about? Right, carbon dioxide is not produced, so 11, this one here is going to be this. Lactic acid can cause cramps, so hopefully everyone got that. I have to put the glasses on, otherwise I can't see jack. I can't see the chat, that's definitely sure. Oh, 11 is B. 12, what is the name of layer A? Layer A is the palisade mesophyll, so 12 is B. 11 and 12 are B peeps. Spongy mesophyll is this layer here. Photosynthesis occurs in the leaves. Which substance is needed for photosynthesis is A. And 14, this is the waxy cuticle, D. So the correct order of the answers was B, B, A, D. Yeah. Blick Biz, the upper epidermis. Okay, thank you for showing up, Lucas. The upper epidermis is not the waxy cuticle. The waxy cuticle is a waterproof, lipid-based, fat oil-based substance that is produced by the cells of the upper epidermis. So it's like the clothes on top of the upper epidermis. It's a chemical that it produces, a fat. The upper epidermis is transparent. They're colorless, transparent, and they allow light through into the palisade cells. But they also produce the waxy cuticle, which waterproofs. Why do we want to waterproof? It's like reverse waterproofing. We want to prevent the water escaping because it's needed in the palisade cells for photosynthesis. Okay, let's move on. We need to smash through because I'm getting tired. Oh my good gosh. It's late. You guys might want to get sleep. Where are we? We got down to gonorrhea. We did gonorrhea. We were on white blood cells. Oh my gosh. Quickly, can you name the 3 ways white blood cells fight disease? Well done, Gold. Phagocytosis, antitoxins, antibodies. Yeah, absolutely. You're right. So phagocytosis is when, like this, you got number 1, phagocytosis. You need to know that white blood cells, they come in all flavors. Look at all these flavors. White blood cells ingest pathogens and digest them by phagocytosis. You can use the word engulf or ingest, and then they digest them. Excellente. The second way, and those cells, by the way, these cells here are called phagocytes. Oh, come on, Tom, you wally. So these cells are, and these cells are phagocytes, but this bad boy here is a lymphocyte. So that's just to show you the difference. That's a lymphocyte here. Lymphocytes produce antibodies that destroy pathogens. And then the third is another type of lymphocyte that produces antitoxins. Now, they're only going to work against bacteria because they're going to neutralize the toxins. What do you want to know about lymphocytes? Lymphocytes make antibodies specific to the pathogen's antigens. The antibodies destroy the pathogen. Your immune system makes memory cells, long-lived lymphocytes that can kill the correct antibody to kill the pathogen. So a lymphocyte is, you have these things called, I'm just going to call them B cells, but if you want, you can call them lymphocytes. Now, what they do is they make a specific antibody, and all you need to do is know that that will kill the pathogen. It will lock on and kill pathogen. So antibodies kill pathogen. However, they are specific. The antigen for killing the Salmonella bacteria will be different than the antibody for killing the Salmonella bacteria will be different than the antibody for killing the gonorrhea bacteria. Okay, so they're going to be completely different. Okay, we just did this, so you should know these. That's right, Ishul, they're complementary. So the answers here, hopefully you can get these, guys. They ingest pathogens. They do not produce antibiotics. They produce antibodies. Antibiotics are a medicine that we can take to kill pathogens. They produce... Oh, twice. We don't want that twice. They produce antitoxins. There you go. They don't produce vaccines and they don't stop pathogens entering your body. So those are the three. I don't know how that happens. Okay, let's explain. Well, no need to explain. We've already got it here. So I'm just going to put this up. If you have any questions, just ask me. I am going to just leave this for you to digest. So if you want to know how vaccines work, this is the explanation. What the fudge? What the floomin' hoobaloo-boosh? No, that's not good. How do we change that? So, vaccinations, how do they provide immunity? Vaccines contain dead or inactive pathogens or weakened pathogens, or they can contain the proteins or the antigens of the pathogen, so part of the pathogen's body. Your white blood cells produce the correct antibody to kill the specific pathogen, and that is what you got to know. That white blood cells produce antibodies. That's what your immune system does, and they kill a specific pathogen. Also, beyond the spec, you can know that they also appreciate this in the mark schemes. You make memory cells, which are just the same as the cells that produce the antibodies. They're long-lived B lymphocyte cells, and they can also make the correct antibody that will kill the pathogen if you come across it a second time. Lymphocyte are a type of white blood cell. Absolutely, the Grid Box podcast. Lymphocyte and phagocytes are the two types of white blood cell. In the AQA spec, you specifically have to know that some white blood cells do phagocytosis. They're actually called phagocytes, but for some reason they don't put that on the spec, but you have to know the process they do is phagocytosis. And then you have to know that the other type of white blood cell, the second type of white blood cell for GCSE, there's actually like loads, but that is a lymphocyte, and lymphocytes are known as B cells or B lymphocytes. B because they're made inside your bone marrow, so it's their location. Okay, they're made inside the bone marrow. Now B cells, B lymphocytes, they produce antibodies or antitoxins, and yeah, antibodies kill pathogens, antitoxins neutralize toxins. I haven't gone through monoclonals. You can just call them B cells or lymphocytes. I can't remember if it's specific. I think it is. Antibiotics discovered by this dude. You need to know that, Alexander Fleming. So make sure you know that Alexander Fleming discovered antibiotics. And the first one he discovered was penicillin. He discovered it from mold. So you do need to know, I didn't write it on here for some reason, but you do need to know he found it in mold. By accident, but you know, it wasn't really an accident. So I have put from mold, yeah. So you do need to know that. Alexander Fleming discovered it, found it in mold. Antibiotic resistance. So what is antibiotic resistance about? Bacteria, how do they get antibiotic resistance? Some bacteria contain a gene. The antibiotic, when you take an antibiotic, it kills all of the bacteria except the resistant ones that have the mutation or the resistant gene. If you don't kill them all, and some survive. The resistant ones survive. They're going to grow back a resistant strain, which is dangerous because the antibiotics won't work on them, or you'll need double the antibiotics, and that's just a never ending cycle. Now let me show you how that business works. So let me just quickly talk you through antibiotic resistance. OMG. All right, let me show you antibiotic resistance. Right, let's imagine this is a person. All right, you might be asked, and it is on both specs. Let's imagine, oh my gosh. Now you can see why I got a level 4 in GCSE. I think, I always hated the examiner, but in hindsight, I think they were generous, if I'm being honest. Right, let's imagine we got bacteria. These are green bacteria. You get the idea, right? Lots of bacteria inside the person's body. They're very sick because of these nasty bacteria. However, some of these bacteria, some of the bacteria, these purple ones, have a gene that makes them resistant. So these bad boys, some bacteria have a resistant gene. Now this person takes their antibiotic pill. I mean, what the hell is that? This is supposed to be an antibiotic. Jeez Louise, this is some bad artwork. So they take their antibiotic, which goes in there, in the old mouth, and it kills, but they don't finish the course, right? They're a bit naughty, and they only take the first day. Which ones are going to die? The weak the normal ones are going to die, and the resistant ones are going to survive. Okay, 3 days in, there's none of them left. Now, they're supposed to take it for 5 days, but they only take it for 3 days. I feel so much better. Oh, I'm not going to take the rest of the antibiotics. Big mistake. What happens is these do binary fission, but because they've got identical genes, now the person gets really sick, and this spreads to somebody else, and now that person has got it. They've created a resistant strain that is now going to go from person to person and is very hard to kill. So we've now got to go back to the doctors and say, take 10 days worth, double strength. Again, so you see the problem. So antibiotic resistance comes around because of mutations. A mutation in a gene creates a resistant strain. All the normal bacteria die, the resistant ones survive, they reproduce and pass their gene on, creating a resistant strain. Okay. So you need to finish. You don't need to talk about T lymphocytes. You don't need to talk about T lymphocytes. They come from the thymus. Okay, I'm going to get on to monoclonals. Foxgloves are these pretty flowers. You do need to know 2 things about them. Foxgloves, 3 things. Foxgloves produce the medicine digitalis that treats heart conditions. And this is part of the spec in both combined and triple. You need to know that foxgloves give us the medicine digitalis, and it treats heart conditions, especially ones where your heart's out of rhythm, the beat. Aspirin comes from the willow tree. You need to know that. You need to link the drug aspirin from the bark in the willow. Okay. Let's go through a bit of drug testing, and then I'll do monoclonals. Yeah, I'll do MABs after this. I'll do this. Drug testing, preclinical trials. What do we... So you have preclinical trials. In preclinical trials, what do we test the drugs on? We test the drugs on animals and cells. So just try... It's the exam's tomorrow, so just remember 3 parts of the drug testing. We test the drugs on animals and cells or tissues, like blood cells. And we're looking for toxicity to see if it's dangerous. Then we move to the first set of human trials. These are clinical trials on people. And we're going to do it on healthy people first. You don't want to give sick people the drugs in case there are side effects and it kills them. So we give the drug to healthy people, normally young, healthy, fit people. And half of them get a real drug and half get a placebo. The placebo is to find out if there's any psychological effects. Okay, so if we just give them all the real, if we give them the real drug, we don't give some people, if some people told they're given a drug, they think they're feeling better even though they haven't been given a drug. So it's that kind of thing. We're trying to weigh up, is there some psychological effects playing up, because humans get affected by these things. So we do a blind trial where the patients don't know if they're on the red pill, the blue pill, the one with the drug in, the one with no drug in. A placebo is a drug. No, a placebo is a pill with no drug in it. Okay. Yes, toxicity, dosage, and efficacy. Absolutely. Efficacy and our 794 just means does it work? So you don't have to write efficacy in the exam. You can just say does it work? Why do we test drugs? We want to know, is it dangerous, toxic? We need to say toxic. Will it, like, does it have side effects? Dosage is how much you have to give people so that they get better, and efficacy is does it even work? Yes, Ishul, well done. We have to do peer reviews. Now, what is double blind? Double blind means that we don't tell the doctor. Why do we not tell the doctor? We want to avoid bias. So, doctor and patients do not know who has, see, it rhymes. The doctor and the patient do not know who has the placebo. Ha 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 ha. Right, yeah, doctor and the patients do not know who has the placebo. So it's double blind. Why do we do this? This avoids bias. So you need to know this avoids bias. You know, maybe the doctor, hello, has taken a small little bit of cash, 500 quid in the back pocket. The drugs company are like, listen, doc, we need you to make sure these results look good for us so we can start selling our drug in the chemists. There's all sorts of other reasons that you want to avoid bias. The doctor might give away who's on the placebo. He might give someone the red pill and go, hey, you're on the red pill. The person says, oh, my head feels better. Yeah, right, you're an idiot. No, it doesn't. So we don't want the doctor to know when he's giving out the pills or she's giving out the pills who's on the placebo, who's on the real drug. But someone has to know, otherwise the results are useless. So the scientist back at the lab is the person that knows. The doctor records the results under red pill, blue pill, passes them back to the scientist, and they know. Anyway, enough of that. Any questiones? Yeah, finally you test on sick people. You test on the healthy people in small dosages as well. Okay, so you kind of find toxicity when you test on the animals. You're going to find out if it's toxic. You don't want to give a drug to humans without knowing if it's toxic first, right? We want to know, is it toxic? Toxic means, is it causing serious cell damage? Does it damage cells? We're going to find that out when we test it on cells and animals before any humans. We don't want to give toxic stuff to humans. That's poison. Kill them. So we find that out in the animals and the cells. Then we go to the humans and we look for side effects. Things like, oh, I get a bit of a headache, or, you know, that kind of thing. Or I feel a bit sick when I take that drug. So we start recording side effects on healthy people, and so that we know it's safe. Let's say some of those side effects could still be dangerous. Some of those side effects, you might get away with saying toxicity on the healthy patients as well. But generally that's the animal cells and stuff. And then when we go to sick people, we're seeing, does it work? Does it make them better? Does it cure the disease or the thing that they're sick with? Now, the final thing, we need to do it on lots of patients, and it has to be a double-blind trial. But there are some ethical things. Okay, let's have a look at some questions on this, and then we will go on to monoclonal. Oh my gosh. Okay. In a double-blind trial, who knows which volunteers are given the drug and which volunteers are given the placebo? And hopefully you know the answer here. Neither the volunteers nor the doctors. Nobody knows. The scientist knows, so don't worry, because that can be confusing. Which bacteria and viruses make us ill because they produce... Actually, that's not really true. Only bacteria produce toxins, so that's an old spec question. Antibiotics are medicines that kill bacteria. Use the correct answer from the box to complete the sentence. Mutate. So, okay, let's not hang around on that too much. That's an old spec question. What's in a vaccine? Small amounts of dead pathogen. You're not going to use large amounts, and you don't want them to be live. They did make some mistakes in the past where like 20,000 people died in America. I can't remember which pathogen it was, what disease it was back in the 1960s or 70s. Or 40s maybe. They injected a load of people with live pathogens by mistake. Okay. Let's just sing through, man. The use of antibiotics. Which of the following is an antibiotic? Hopefully you know this. It's penicillin. Why have antibiotics not reduced deaths to 0? Yeah, I'm going to get onto that. I'm going to quickly do monoclonal, then I'll do bioenergetics. Yeah, I'm going to shoot off of this because this is like I'm flaking. Okay, so the reason is antibiotic resistance. What does that mean? That means that some of the bacteria have become immune. They're not immune. That's a bad word. Naughty, naughty. You mustn't say immune. Immune can only happen if you have white blood cells. Bacteria are a cell. They don't have white blood cells. So they can't produce antibodies. Bacteria become resistant due to mutations and natural selection, which I talked about earlier. So antibiotic resistance. The other thing is people don't finish the course. People don't finish the course. So some diseases are, I'm just giving an example, are caused by viruses. Or what we can say here is antibiotics, oh my days, only kill bacteria. Okay, they don't kill viruses. They can't kill viruses. Right, let's get off of this because this could just go on and on and on because I have hundreds of these questions. No, I'm not going to do these, man. I ain't got time for this kind of business. Where are we going? Monoclonal. Let me quickly just run through monoclonal antibodies. Or I'll fall asleep. And then bioenergetics. Jeez Louise. This is the last thing I'm doing on this topic. Okay, monoclonal antibodies for them trips people. Let me just zang through this. Okay, let me zang through it for you. Bosh. Right, what we're going to do. Oh, Lord have mercy. This is wiping man out. So what we're going to do, number 1, inject mouse. We're going to inject the mouse. Okay, we're going to inject the mouse with, oh my lord, inject mouse with protein. So I'm injecting the mouse with protein. It could be an antigen, it could be whatever we want to make a complementary antibody for. So inject the mouse with a protein. The mouse produces a B cell. You can call this a lymphocyte. So, or you can just call it a B cell or a lymphocyte. The mouse produces a lymphocyte. That makes an antibody that is makes complementary antibody. Now, if I come up here, what you might be going like, I don't get it, sir. Right, the protein that I'm injecting is going to be a specific protein that I want this to join with, right? This can join with the protein. What do I mean by protein? It could be the antigen of a disease. If I want to test if you have HIV, it could be a protein that's produced when you're pregnant called the HCG protein. It could be a protein that's produced when you take bodybuilding steroids and comes out in your blood. That's how we catch athletes and boxers who cheat and take certain chemicals. So proteins are get produced in your body. If I want to find the protein and identify it, I need to make millions of these antibodies so that I can use them in test kits. Now this is just explaining how we make monoclonal antibodies. It could also be a cancer. Maybe this is a protein produced by lung cancer cells. So I want to make monoclonal antibodies that will only lock on to lung cancer cells, and then I can attach something to destroy the lung cancer cells. Anyway, you get the idea. This stage is about how do we make the monoclonal antibodies, not what they're used for. So I'm going to inject the mouse with the protein. The mouse will produce a B lymphocyte that makes the correct antibody that's complementary to that protein. Then what I need to do, B lymphocytes reproduce slowly. So I'm going to get a tumor cell. Here is my tumor cell. Notice the shape of the tumor cell. They often show it in diagrams and things as having a wibbly wobbly cell membrane. Okay, I'm going to get my tumor cell and I'm going to fuse with a tumor cell. So what I'm doing, this would be number 1, this would be number 2, makes a B lymphocyte, then I fuse it with this tumor cell like so. Now when I fuse it, to show that I fused it, I'm still got the wibbly wobbly tumor cell, but I've now got the nucleus of the white blood cell. I don't want that bit of the cancer cell because that's not going to make the antibody. This has the genes for the antibody. So now I have this, and you might say, well, why did you do that? That's a bit dumb. Well, these reproduce slowly, slow mitosis, or kind of normal mitosis. This does uncontrolled growth. Remember from organization chapter, uncontrolled mitosis, rapid cell division. So I am going to very quickly have 1 million of these, so billions of these, each one producing billions of antibodies. I have gazillions of antibodies that are identical. Now we call this cell a hybridoma. Where's my pen? This cell is called a hybridoma, and it reproduces rapidly. Oh my good gosh, making lots of monoclonal. Monoclonal means the same clone, one clone. They're identical. These antibodies are all identical, so it makes lots and lots of monoclonal antibodies. That's the hybridoma there. And lastly, he's run out of room, number 5, I want to separate my monoclonal antibodies from these cells. I don't want these cells on my testing kit. I don't want these cells at all. They're just there to make the monoclonal antibodies. So I'm then going to just say filter and purify. And then I now have my monoclonal antibodies. Yeah, this is separate only. This is separate only. Now, if you're asked about what the monoclonal antibodies do, monoclonal antibodies can bind to specific proteins and we can attach a medicine to them that will kill cancer cells or something like that. They can also be used in testing kits and stuff like that. Yes, you fuse with a tumor cell. Yeah, hold on. Oh, geez, how do I do that? Hold on, how do I go new? All right, I want to get onto bioenergetics. I actually, oh my gosh, because I'm never gonna finish. Are we never gonna finish? Am I never gonna finish? Right, so essentially with monoclonal antibodies, now I have my monoclonal antibodies. Let's imagine this is a lung cancer cell. That's probably not the right name. It should be called a tumor cell, which causes lung cancer. Now, let's picture these are the proteins that are on lung cancer cells. What we're saying is, if I have my monoclonal antibodies, I can attach a medicine. Now, the healthy cells in the lungs, let's call this healthy lung cell. The healthy lung cell has a different shaped protein, so this cannot kill that cell, but it can lock on because it is complementary. The shape of the antibody fits with the protein, and so that way we can just target the lung cells with our monoclonal antibodies and they will lock on and destroy those lung cells. So that's the theory. Now, we can also use it in testing kits. In your blood, if you have COVID, you will produce certain proteins, and we can attach coloured beads, and you can wee on the stick or stick it up your nose, whatever you do with the different sticks, if your pregnancy stick, and the beads will all line up together if they attach to the protein. The main thing is just to know that the monoclonal antibody is identical clones of each other. They are complementary only to the specific protein in the question, and therefore they will attach. Now, we can also say that if monoclonal antibodies attach to certain cells, they'll stop them reproducing or they'll kill them. The drug will kill them. Okay. Right, let me just go to don't worry, the thing is, let me go bioenergetics, because we're going to be oh my gosh, this has got, I've gone through so flaming slowly, man. I had so many questions to go through. Never mooned. We all got history in the morning, history exams. What we got. Oh my good gosh. Let me speed through bioenergetics. I think I'm flaking. Okay, bioenergetics, you know the coup. I'm just going to go through quickly. So, bioenergetics, you have carbon dioxide, 6 carbon dioxide, and we're going to have 6 H2O. Let's use a pen that actually stands out. 6 of that. And that is going to produce 6 O2. And it is going to make C6H12O6, which is glucose. And glucose is then used, so that's photosynthesis, and we need sunlight, and we need sunlight energy. Oh my gosh, what the hell is that? What the blah? We need energy going in, so light energy, and that's photosynthesis, right? Ta-da! Complete the word equation. Describe how the energy for photosynthesis. So that's easy. I'm not going to do that. That's just light energy is absorbed by chloroplasts or chlorophyll. What you do need to know specifically is how do plants use glucose. You need to know 5 ways that glucose is used. So I'll go through these very quickly. We're going to say respiration, both anaerobic and aerobic, for both plants and animals and fungi and all living things. Glucose can be joined. Glucose plus nitrates makes amino acids, makes protein. So glucose and nitrates join together to make amino acids and get turned into protein. Protein's used for growth. You also need glucose for doing any metabolic reactions like joining stuff together. Fats, lipids, oils. So in a plant, the plant will turn it into waxy cuticle, lipids for the waxy cuticle, or lipids for the cell membrane. And then cellulose is made of glucose. That's right, cellulose is made of glucose. So when plants make glucose, some of it is used for cellulose in cell wall. Oh gosh, what happened there? Yeah, it's all going a bit pear-shaped here as I'm flaking out. And for making starch, which is long-term energy storage in the roots. There we go. You could also say chlorophyll. Glucose is needed to make chlorophyll. Okay, Blick Beers, you get 2 lines because one is a control line. So essentially you have 2 lines. One line has immobilized antibodies that are complementary. The control line has special antibodies. They're different, a second type of monoclonal. So you have a monoclonal antibody that is complementary to the protein that you're looking for, like the HCG. And then what you have with the control line should always go blue, even if someone's not pregnant, because it has monoclonal antibodies that are complementary to the blue bead. Or you can say not to the blue bead, but complementary to the other monoclonal antibody. So it's a bit complicated, but essentially when you wee on the stick, monoclonal antibodies will move up the stick. These are ones with a blue bead attached. We then cleverly have made a monoclonal antibody that is complementary to the first antibody. So as it goes up the stick, they will always stick to that one and light it up blue, regardless of whether you're pregnant or not. Whereas the first strip, the one that means you are pregnant, will only light up if the protein is present. The second one will always light up, and this just proves that the strip is working. Because if the strip isn't working, neither would it might be that the monoclonal antibodies haven't moved up the strip, and so it proves that they've moved up the strip and they've moved past the test area. Anyway, that's long for me to explain for the value of it probably not coming up. Okay, so for example here you could be asked, give plants use glucose for respiration, give 4 other ways they use glucose, check the answers: to make storage, to make fat or oils for storage, starch for storage, to make amino acids, proteins or enzymes, and to make cellulose for cell walls, and then any other named way for active transport, for making chlorophyll. You get the idea, peeps. Now, there are 3 limiting factors for photosynthesis. So the 3 limiting factors are, you have light, carbon dioxide, and people think water. It's not water, it's temp. Don't write temp, write temperature. Okay, and then on the side I'm just going to write P, which means the rate of photosynthesis, because I'm being tired now. So light, as I increase the light, photosynthesis goes up. And what this tells me is that position A, light is limiting. If you give somebody something more and it goes faster, it means they needed that thing. So if you own a cookie shop and you're making cookies and I deliver you more flour and you make your cookies faster, it means you needed flour. But if I give you more flour and you don't make the cookies any faster, it means you didn't need flour. It's not flour that's limiting. So on this graph here, as we move across the bottom, I'm giving it more light, and guess what? Photosynthesis is going up. That means that at A, light is limiting. But at B, I'm giving it more light, but it's not increasing. So light is no longer limiting. We then have to suggest what is the limiting factor. It's one of the other two. So it's either CO2 or temp that is limiting. And that's how you've got to read these graphs in the exam. Now for carbon dioxide, you're going to get the exact same shape. You're going to get a shape like that. And it's the exact same thing. So at A, CO2 is limiting. And I know it's limiting because as I increase the carbon dioxide, the plant starts growing faster. It photosynthesizes more. That means it needed carbon dioxide. But at B, I try to give it more carbon dioxide and it's like, I ain't growing any faster. That means at this point, it's got enough carbon dioxide, but it's either the temp or light is limiting. And then we have a bit of a different graph for temperature. It's a bell graph like this. And this one just shows that here at A, and B, and I've kind of got a C as well there, which I'll talk about. But A, yes, temp is limiting. But at B, we'd say because there are enzymes in living things, if we go to higher temperature, you just have to know enzymes are denatured, and that means their active site has changed shape and no longer fits with the substrate. So you don't get the substrate, and the enzyme can no longer bind, or the substrate cannot bind into the active site. By the way, this is going to be the optimum temperature. That is known as the optimum temp for photosynthesis. Okay. Oh, chlorophyll can also be a limiting factor, which is the green pigment. Okay, I don't want to spend too long. Let's do the practical. What can I say? POMWID practical, guys. You need a stopwatch. Whenever we say rate, you think time. Rate is always something divided by time. If they say, how would you calculate the rate of photosynthesis, the rate of respiration? You're always going to divide something by time. How would you calculate the rate of mitosis growth or the rate of growth of cells? You do something divided by time, right? So always you're going to need a stopwatch. Here, we could count the bubbles of oxygen, and they often do that. But remember, oxygen is given out during photosynthesis, but that's not a very accurate way to do it. It's much better to use this thing over here, and this is called a gas syringe. A gas syringe actually measures the volume of oxygen given out accurately. And the reason why bubbles aren't very good, you need to be able to say bubbles are different sizes. Therefore, it's not accurate to say, oh, it gave off 4 bubbles. That's the same as these 4 bubbles. No, it's not. That is a lot less oxygen than these 4. So the fact that bubbles are different sizes means that is not an accurate way to measure the rate of photosynthesis. To measure the rate of photosynthesis, we should do volume of oxygen over time. Okay. Now, for photosynthesis, we need to keep some things the same each time, and we need to change one limiting factor and keep the other limiting factors the same. So, for example, if I want to investigate light, I'm going to keep the distance of the lamp from the pondweed. I'm going to change that using a ruler. And if I want to change CO2, you use something called sodium hydrogen carbonate. Let's have a little look. Hold on. Let's not do that. Let's just go through and explain this. Okay. I'm going to make that big. Gosh. Right. So, if IV is CO2, if what you're changing is carbon dioxide, then keep temp same using a water bath. Don't say a thermometer. That won't keep anything the same. That just measures. Use a water bath to keep the temperature the same so that I can investigate how CO2 affects photosynthesis. And then I would say keep light intensity the same. So keep light intensity the same using lamp and ruler. Same distance. Oh my gosh, this is horrific. So keep the temperature the same with a water bath, change the light with a ruler, or keep it the same with a ruler. And then for CO2, you've got to use something called sodium hydrogen carbonate. Okay, and if you adjust the amount of sodium hydrogen carbonate, it will change the amount of CO2. You need to know that. And if you want to keep CO2 the same, then you keep the same amount of sodium hydrogen carbonate each time. I have got a live where I went through all the core practicals, to be honest with you, but let's have a look at this little questionnaire, right? So we're going to skip all of this kablapple. Suggest 2 ways the method could be improved to measure the rate of photosynthesis more accurately. Have a look, people. What should they use? If we want to measure it more accurately, rather than counting bubbles that are all different sizes, what should we do? We're going to say use a gas syringe. Now, something else we can do, another way to do is to use a lower intensity of light. I hear you say, what? Use less light? That will mean that the bubbles come off more slowly and make it more easy to count. So that's another thing I could do. I could use less light or a lower intensity of light, slow down the release of the bubbles, make it easier to count. Now, the LED light source does not get hot. Why is it important the pond weed remains at a constant temperature? Well, what are we investigating? Are we investigating temperature? If we're investigating temperature, then we want to change the temperature. But if we're not investigating temperature, we need to keep the temperature the same. Why? Temperature is a limiting factor. And look, you can see here, we're investigating the intensity of light. So that's the IV. Repeat the pond weed at different distances. We're investigating intensity of light. So we want to keep, I don't need to say keep temp the same. It's in the question. Why do we keep the temperature the same? We just want to say, we could say lots of things. Temperature is limiting factor. And then I could say like increasing temp increases, oh my days, rate of photosynthesis. So increasing the temperature increases the rate of photosynthesis. Or I could waffle and say that increasing the temperature would denature the enzymes and ruin the experiment by denaturing the enzymes, change the active site, they'll no longer fit with the substrate. Describe how the student could change the method to investigate the effect of carbon dioxide concentration on the rate of photosynthesis. So this is testing. Do you know how to change that? Well, look, they've given you the clue there. So we would say like use different, and you have to know this, but don't write conc in your exam, you won't get the mark. Use different conc of sodium, hydrogen, carbonate solutions. So use different concentrations of sodium hydrogen carbonate solutions. And it says state two control variables. So that is going to give me the mark for explaining how I'm going to change the IV. And then what I would want to say is keep the temp the same. And I'm going to say use water bath. Ah! I'm dying, people. Keep light intensity the same. Oh my gosh, use ruler. Well, I really need to say keep the same distance. Oh my god, I'm dying. I think I am done. Light intensity can be calculated using the inverse square law, where L is light intensity and D is the distance of the pond weed from the light source. The student placed the pond weed at 5, 10, 20 centimeter from the light source. Explain how light intensity changes as the distance of the pond weed from the light source is doubled. Now, before you say, is this triple? This is from a combined paper, so this is actually a triple and combined. That means equal to, is proportional to. So light intensity is equal to 1 over, that's light by the way, that's an L. Light intensity is equal to 1 over the distance squared. So all I want to do is show you that if I go 1 divided by 5 squared, that is equal to me saying 1 over 25. So when my distance is 5, my light intensity, my Li is 1 over 25. If I double the distance, so this is at 5 centimeter, let's double the distance to 10 centimeter and see what we get. You only need to do 2 lots of these. You don't need to do all of them, but if I go 1 over 10 squared, what is 1 over 10 squared? That means my light intensity is now 1 over 100. What has happened to the light intensity? The light intensity has decreased by 4. So it has divided by 4. But I have doubled the distance. So D has been doubled. My Li has been quartered. And that's the relationship because of this. So if I triple the light intensity, if I triple the distance, my light intensity will go down 9 times. Yes. So anyway, that would get me all of the marks. So just showing, and I'd write that, I'd say when you double the distance, you quarter or make it 4 times weaker the light intensity. So based on this, predict how many bubbles of gas would be produced in 1 minute if the pond weed was placed 40 centimeters from the light. Well, look what they did. They doubled the distance. What happened? It got 4 times weaker. Roughly 130, divided by 4. Okay, it's roughly, but you know what, if you times that by 4, what you're going to get? 124. So it's roughly divided by 4. Now look, they double it again and roughly divide 31 by 4. Roughly 8, right? 32 divided by 4 is 8. So roughly each time they're doubling the distance, the number of bubbles of oxygen coming off are getting smaller. When they double the distance, they're timesing that by 2, that's dividing by 4. Timesing that by 2, that's dividing by 4. So guess what? It says, make a prediction. How much would be produced if they now double the distance again to 40 centimeters? How many bubbles will be produced? No unit for light. The light is lux. So double the distance to 40 and this divides by 4. So 8. We're going 2 peeps. There's going to be 2 bubbles. So you're just showing, and you're just showing like when you double the distance, you quarter that, and you can show that by going right. 8 divided by 4 equals 2, and that'll get you all of the marks. Yes. Artificial conditions. This is just saying, listen, if you've got a greenhouse, you might want to put light bulbs in there, a UV light to give the plants light so that they grow faster. You might want to burn fuel in there, paraffin heaters, so that carbon, or put a bit of burning coal or whatever. You put a heater on, let off some carbon dioxide, increase the carbon dioxide level in there. Okay, you do need to know that photosynthesis is an endothermic reaction because it absorbs light. Respiration releases heat, is an exothermic reaction. Is some so nearly done with this. A gardener grows tomato plants. The tomato plants develop yellow leaves. What would be the best way of improving the growth of these plants? So why do they get yellow leaves? What would we add to the soil to improve their growth? So what we're going to do, we're going to add minerals. So that's probably lacking in nitrates or lacking in magnesium. If you do triple, you need to know that magnesium is needed to make chlorophyll. Without chlorophyll, you get yellow leaves, which is called chlorosis. Tomato growers alter the conditions in greenhouses to make tomato plants grow faster. Which changes would make them grow faster? Increasing the temperature. Yeppy doodle. It's one of the limiting factors. Increasing the oxygen concentration. Increasing nitrogen. Turning the lights on at night. That will allow it to do photosynthesis all night long. So that's just, nitrate is good for growth gold, and a lack of nitrates gives you yellow leaves. A lack of magnesium gives you yellow leaves. Aerobic respiration happens inside the mitochondria. It's glucose plus oxygen makes water and carbon dioxide and lots of ATP. It's exothermic. Put your hand under your armpit, you'll feel the heat coming out your body. Aerobic respiration is exothermic, whereas photosynthesis is cooling. It absorbs and takes in heat, so it's endothermic because it takes it in from the sun. Okay, you do need to know this, and this is one of those that you can just repeat all the time. Why do we breathe faster during exercise? So you need to be able to smash that out as to why we breathe faster during exercise. And so why we breathe faster is We breathe faster during exercise because we need more O2 for aerobic respiration. Why? Because we need more energy for muscle contraction. Okay. Yeah, more or less. We're getting onto that. Yeah, increase rate. A little bit of blur. Absolutely spot on. Yes. Yeah, on the last bit. We're nearly there. Hallelujah. I mean, I had about 300 questions, which I didn't go through, but that's because I was just waffling all night. Oxygen from the lungs is carried by our blood to the cells in our body where aerobic respiration takes place. Complete the two spaces to balance the chemical equation for aerobic respiration. So we want to do glucose C6H12O6 plus oxygen goes to 6CO2 plus 6H2O. Name the substance with the formula D. That's glucose. Name the structures in the cytoplasm. That's mitochondria. Okay, lovely. Let's move on, folks. We're going to look at anaerobic respiration. So in muscles, if you've got the muscles, so anaerobic respiration is going to turn glucose straight into lactic acid. What's the problem with lactic acid? Pain, toxic, fatigue. If you ever look at a boxing fight, after the more muscly the boxer, after about 3 or 4 punches, or after about 2-3 rounds, they just get exhausted because all of those muscles fill with lactic acid and become sore and tired. Okay, that's why. Yeah. Anywho, it's toxic, causes oh yeah, I've already put the answer there. Where does lactic acid get broken down? It's in the liver, so you need to know that. Lovely jubbly. When does the body do anaerobic respiration? During intense exercise, when oxygen cannot be delivered to the muscles quick enough. Does it release as much energy as aerobic? No, only releases a little. So you might go, sir, this is what kind of mashes people's brains up. Sir, if I'm having to like lift up a big weight, why would I only need a little energy? Because that's going to use anaerobic. You're still using aerobic. It's just you've reached the maximum. You can't deliver any more oxygen to your muscles. You've got to go breathe it in. It's got to get pumped by your heart and get to your muscles. So what you're doing is you're saying, muscles, use all the oxygen that you've got there, and then you've got extra glucose without oxygen. Just smash that glucose up and make a little bit extra energy and give you that little bit extra. The downside is that you produce lactic acid, which is toxic. We need to break the lactic acid down, and you do that when you've finished exercising, and that is the amount of oxygen you need to break that lactic acid down. The volume of oxygen is known as the oxygen debt. So why do we breathe harder and faster after exercise? Nice and easy to explain why you breathe hard during exercise. We already went through that. You need more oxygen, more aerobic respiration so that you can contract your muscle, so you can release energy for muscle contraction. That's during exercise. But when you've finished running, why are you breathing harder after you've stopped using your muscles? And the reason is why your heart rate and your breathing stay higher is because we need the exact volume of oxygen in to convert the amount of lactic acid we made into harmless substances, into glucose or carbon dioxide and water. And this is done in the liver mainly, but also in the muscles. And we break the lactic acid down into less harmful substances, but we need the oxygen, the exact volume of oxygen. So if you need 50 centimeters of oxygen to break all your lactic acid down, your oxygen debt is 50 centimeter cubed. If you need 300 centimeter cubed, your oxygen debt is 300 centimeter cubed. So that's oxygen debt. It's the amount of oxygen you require to break down the lactic acid you produce during the exercise, and it's why every human being breathes hard after they run. Anaerobic respiration in plants and yeast is different. Yeah, so if you walk down the street, you're not going to do as much anaerobic respiration, so you're probably not going to get as sore. But listen, you're like just sitting on my chair like I am, I'm sore because the muscle has been in a certain contracted position for long enough, stabilizing me, that it's producing some lactic acid. So you like, not a lot, but you will be producing lactic acid all day, and it will need to get broken down. Okay, what's another name for this process? So glucose to ethanol and carbon dioxide. Okay, so glucose to ethanol and carbon dioxide. I have written carbon dioxide, but it's there and it's kind of gone funny. So anaerobic respiration in plants and yeast. They don't make lactic acid. They make ethanol and carbon dioxide. And we can call this fermentation. And it has two uses. The CO2 makes bread rise. And I know what people are thinking. Does that mean my bread has alcohol in it? Can I get drunk off eating bread? No, you can't, because alcohol evaporates in the oven when you make bread and but goes away. So the bread is left behind. But the carbon dioxide makes pockets and makes your bread spring up and get all fluffy and nice. Now, how can we use it to help us in the industry? So for that, and then the alcohol can be used for drinks, for alcoholic drinks. So the ethanol is alcohol. And so you can use it to make alcoholic drinks. I mean, you can use ethanol in hand wash and other things. It's great at killing bacteria and stuff like that. There's one more thing on this. There is metabolism. I can't be bothered to go through metabolism. So what is metabolism? Metabolism is taking small molecules like amino acids and joining them together to make big molecules called proteins, and that requires energy. All of the chemical reactions happening in your body, the rate, the speed at which they happen is known as your metabolism. So right now as we sit here, your body is taking some protein and chopping it up into amino acids, and in another part of your body some amino acids are being turned into protein. In some parts of your body, glucose is being joined together to make glycogen and being put into muscles for storage because it's insoluble, doesn't affect osmosis. And in other parts of your body, glycogen is being chopped up and turned into glucose so those parts of the body can respire. And in some parts of your body, starch is that you've eaten, because we don't make starch in our body, but it's being chopped up and turned into glycogen or glucose for respiration. And you get the idea. You're depositing fats around your body. You're putting fatty acids and glycerols together, and you're making fats to insulate your body. So we've got all these chemical reactions happening every second, trillions of them in our body. And they're known as your metabolism, and they can be ones that build molecules or ones that break them down. And all metabolic reactions are controlled by enzymes. So yes, most of the enzymes we focus on in GCSE are protease, lipase, amylase, the ones that break molecules down. But guess what? There is an exact enzyme for building, and it's not protease, that's a breaker. But if you're joining there is an enzyme that joins amino acids together. You just don't need to know it. There's an enzyme that joins glucose together to make starch. You don't need to know it. We just focus on the ones that digest, but we have an equal amount that are building molecules. So metabolism is about building molecules and breaking them down, and you just have to have a general understanding of that. We already did that. Okay, yeah, I think, folks, always are gonna finish. I'm about to shoot off, folks. Yes. So, aerobic respiration. Type of respiration in human cells, aerobic. Does it produce ethanol? Does it use oxygen? Does it use carbon dioxide? Does it produce lactic acid? Hopefully you know. Aerobic respiration uses oxygen. Anaerobic respiration in humans produces lactic acid. It doesn't make carbon dioxide and ethanol. That would be in plants. The table below shows the amount of energy released by aerobic and anaerobic respiration. Have a look. It is a lot more, 16 compared to 1.2. So it's like 14 times more. Suggests why human cells might respire anaerobically even though only a small amount of energy is transferred. Well, we might if there is not enough oxygen to meet demand. So it's not that our cells wouldn't prefer to do aerobic. Of course they want to do aerobic. It makes loads more energy. But there are situations where there just isn't enough oxygen to meet the demand. You're going to run the 100 meters the oxygen cannot get to your cells quick enough, so your body says, listen, I'm already doing aerobic, but let's tap in and do some anaerobic and get that extra bit of energy to go faster. Okay, but there's consequences. Now, yeast is used in brewing and baking industries. Why is yeast used in these industries? So this is just really, you're going to write out the equation for ferment. So you're going to get a mark for saying like, oh, fermentation. And then you're going to get a mark for saying glucose goes to carbon dioxide plus ethanol. And then you're going to say CO2 used to make bread rise. And then you're going to say ethanol used, oh my gosh, run out of room, in alcoholic drinks. So notice we have written the equation as well to get those extra marks. All right, peeps, I'm tired. I got to go to sleep. I'm smashed. Good luck, guys. Remember, I've done lives that are more detailed than this on each individual topic. You can check them out in the morning if you have time. You can bunk your religious studies exam. You can knock history on the head, and you can watch those videos instead, or you can not. I'll see you later. Make sure you don't spread disease, and yeah, take care. Good night. And good luck. Make sure you guys smash the exam.
I've had training.
The training is nothing. The will is everything.
The will to act. Yield.
Yeah. Cool, guys. Nice one. Yeah. Take care, guys. Just give you all a quick boom for showing up. Boom! Benign and malignant tumours. I'm not going to write it down now. My hands are gone. Benign and malignant tumours. Benign tumours. Benign tumours do not spread and do not invade other organs. Malignant tumours spread and invade other organs. They break off and move around your body and spread and invade and damage tissues and organs. Both grow uncontrollably, but malignant tumours grow at a faster rate. So really, if you're comparing them, they both involve uncontrolled mitosis, unwanted like mitosis, so they're growing faster than normal. And malignant tumours form cancers. Benign tumours do not form cancers. Malignant tumours break off and spread around your body. They travel from like your lungs to your liver or your kidney. They can go from one organ to another, and they invade and damage organs. Benign tumours are the good ones, if you can call them good. They're not really good because they could become malignant at some stage if exposed to radiation and other things. All right, guys, don't forget, you have been amazing. Okay, yeah, that's a little bit dodge. All right, see you a lot later. Take care. Good luck. I'm going to stop being an idiot now.
Whenever I'm about to do something, I think, would an idiot do that? And if they would, I do not do that thing.
Okay, I'm off. Take care. Good luck.
You're still here? It's over.